Yeah, now I think we are good to go. Thank you so much once again for joining. So we were only discussing the reason and the morale behind coming up with this topic. So in the last two classes, there was a question that was raised regarding taking us through why are we doing alignment? Why are we doing using Eugene? Why are we doing this? Why are we doing that? So I will try to take us a little bit before we go deeper into. It will be like a primer into the discussion on career prospects and winning grants and scholarship in South bioinformatics. So I would begin by sharing my screen, uh, my entire screen. So luckily, I am presently in the lab. I think you should be able to see my screen. Yes, I think my screen is now visible. So I would begin by sharing, I will begin by opening my first publication using bioinformatic tools. So this, Asha Wakintola. So a simple search would bring about some of my latest information. So let us go to ResearchGate that houses my publications or Google Scholar. Okay, so the first one I would like to take us through is the my first publication on getting here. The title was Genetic Diversity of Bemisia tabaci Cryptic Species in Nigeria and their relationship with endosymbionts and acquired begomoviruses. So I worked on three different aspects. One is the genetic diversity of the insect itself. The other one is the bacteria, and the other one is the viruses. Is the virus that affects the plants and also the insect. So now let us check the PDF of the file. I promised to download and share with the group, but unfortunately I forgot. So thank God this opportunity presents itself. So now the work is based on three, and what I used throughout was um, bioinformatic tools, bioinformatic analysis, which is genomics, and which is the simplest part. Don't let me say simplest. It's one of the preliminary stage. The first stage is whereby you say you want to do a bioinformatic analysis. So I hope this is at least this is visible to a certain extent. So this shows the region from which I collected my insect samples. And then this is a tabulation of the sample ID, the site, the location coordinates, the date, the host plant, the cryptic species. Now, from the cryptic species Endosymbionts and begomovirus, I was able to denote what happened to the particular insect. This is, for example, this is one insect, and I found out that it belongs to SSA1 SG1 cryptic species, and the endosymbionts that are present is Arsenophonus cardinium, and we have Wolbachia. And the begomovirus that is present is the tobacco collie shoot virus. So this is a tabulation of the summary of all my results. So let me bring people in. So that is how I was able to, that is the summary of my, of each of the samples. I collected in the analysis, I used 23 samples of this insect. Now, this information contains the primers I used to carry out the experiment. So I used 1, 2, 3, 4, 5, 6, 7 primers. I used 7 primers to carry out the experiment. Now, when you Now, this is literally I'm taking us through the process of which to maybe to understand and also to read bioinformatics paper. Now, if you want to carry out your analysis that is similar to what I am about to do, and you are looking for reference materials, so the first place to go is the materials and method. So here, the collection of the samples, normal. And then this is the, for those that are asking about the DNA extraction protocols. So this is my own DNA extraction protocol. If you want to follow this protocol, you just read my materials and method, and then you apply that in your laboratory. Now, this goes to the next stage is the polymerase chain reaction, which is the PCR analysis. So if you read this procedure, you understand the PCR analysis that I passed through, I carried out. Now, this is also, this table here shows the PCR analysis and also the PCR primers. So it shows here that the annealing temperature I used was 52 degrees, and the size of the, after, if I run the gel, is going to be 800, 600, 400, 700, and the likes, and so on and so forth. So this is the PCR analysis. And then the DNA sequence analysis. This is where the bioinformatic analysis work started. So the other ones we described is the molecular analysis part. So now is the uh, bioinformatic analysis section. So in this section, so these are the tools I used. So I used BLAST and I used Cluster W. Now, what do I use BLAST to do? It says here that the gene bank database for the new National Center for Biotechnology Information was searched using BLAST algorithm. So from when I sent the PCR products, after you do your PCR and you run your gel, and you see a gel showing like gel showing the band, then you send that sample to the sequencing company. So the sequencing company now gives you the sequence result. So from the sequence results, so for example, the first one, I do the BLAST algorithm. We all know what BLAST is. So you do the BLAST, you know which particular species your plant, your sample is. And then I use cluster W using bioedit to do the alignment. Now, for those who are novice in research or in what is being discussed, you might not understand fully. You might think this is the whole stuff regarding what I did. But there are hidden messages in the DNA sequence analysis, in the collection, in the DNA extraction, in the PCR, what have you. Now, this, if you are just reading papers and you are just reading it for, oh, you want to get the full gist about a particular analysis, and you are just reading the paper, ladies and gentlemen, you would be there and you will not get results. You will do and do and do and do, and you will not get results. The reason is that I was the one that knew how I carried out this analysis. This is just a summary of the whole picture. This is not the full picture of what I did to do the DNA sequence analysis or to do the alignment. Now, if you are just coming up and you are reading this and you are trying to read and read and read and replicate what is reported here, you would it will take you so long time, so much long time, that you will labor and you will not get the result. And that is where a mentor comes in. I was not able to do all this except from the guidance of my seniors in the laboratory. So they are the ones that took me through each and every process. From the start, I thought, why must it be difficult? Haven't they discussed the whole process in the materials and method? But that is not the case regarding scientific science work like this. There are tons and tons of other information that do not make it to the publication. They do not make it to the reportage that we have here. So you must know that you must have it at the back of your mind. Every paper you read, every paper, except you are an expert in that already. Every paper you read, there is, if it is not 20% reduction in the publications that later make it to the publication, it will be 30%. And that 20 or 30% is very, very germane to you having or replicating the result that you will see them reporting. So that I need to point out that part, especially for every one of us here. So okay, let me quickly download and I will share. So that is one thing, explanation regarding the PCR product or what have you. Let me share the material in the chat box. If it's gonna go, I don't think it's possible. Okay, I would share it on the um, on the WhatsApp group and the Telegram page also. But that is by the way. Okay, let's continue. We can do that later. So now, after doing the alignment, now for those who are asking why are we doing alignment, now you can see the alignment is an integral part of the work. Now moving to the phylogenetic analysis. This is the core of the bioinformatics analysis. This is the first stage. This is the second stage rather. Alignment is the first, then second stage is the phylogenetic analysis. So there are different reasons why we do phylogenetic analysis, and one of such is the evolutionary relationship between your samples. So I aligned all my samples with samples from other samples from Nigeria, from Ghana, from Mozambique, from Tanzania, from Uganda, from Kenya, from Burundi, from Malawi, from Seychelles, Israel, Burkina Faso, Syria, Sudan. So so that I can know to which region or to which uh, uh, what can I mark my samples with? So that is the purpose of blasting your samples. So I merged all my samples with all these samples that are submitted in the NCBI, and I also submitted my samples to NCBI. If you search this accession number MK532703, you will see the full details about this sample, and you will see that I'm the one that submitted and make the other analysis regarding that. Now, that's one stage. Now, this is another result which shows the pairwise comparison of the cytochrome oxidase 1 among my samples. This is another bioinformatic analysis tool. So now, for you, as simple as it seems, this is only one tree. I did one tree, phylogenetic tree, even though there are many other trees that didn't make it to these publications, and several tables. These ones are statistical calculations, so they are also part. And if time permits, we are also going to get to that. So these are different tables that shows. But before I arrived at these tables, I've performed different different bioinformatics analyses in the background, but they did not make it to the publications yet. So, and I have them everything in my thesis published already. So the thesis was all this thesis was summarized into these few pages. So there are a lot of information that go unreported regarding that do not make it. Not that they are not reported. That do not make it to the publication. So that is one thing that you need to have when you want to start out in your bioinformatics journey. You need to have a mentor to put you through. How do they do this? Recently, it's not that long. I think around February and March, I wanted to start learning about metagenomic analysis. It took me and what I did was that I started reading materials on metagenomic analysis. But unfortunately, I was trying to replicate the material. I wasn't getting the results. It took me 2 weeks of little sleep, sitting in the lab. I shut down from a lot of other activities I was doing. I was just focusing on one thing, but still, I wasn't getting results until when even I contacted other people that are in the metagenomic analysis, they were sending me materials. They were sending me materials, but I wasn't getting it. I wasn't getting it at all. I wasn't getting. I wasn't. I did this. I followed the same process. The same process. I did not get the same result. Why? Until one of my mentors in my teachers in metagenomics, he came. He came to my lab and said, "Okay, let me see what you're doing, Ashraf." And he said, "Okay, okay, you follow the same. Yes, 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 yes. Ah, Ashraf, you made a mistake here." And what I've been laboring to do for like 2 weeks non-stop, he did it within 1 hour, not even less than 1 hour. He saw the problem and he solved the problem in less than 1 hour. So that is one thing that you need to know regarding if you want to start out your journey in bioinformatics. So you need to have a mentor in order to save you time and stress. So that is one part of my publication to explain what do we want to achieve with bioinformatics the alignment. Where are we going? What's our goal? Now the second one, I will show you this one, is about fall armyworm. Is a fall armyworm is a pest of agricultural importance, like the first one. So let us view the full text. So this is also my publication, but unfortunately I couldn't complete the work before leaving my lab, the lab where I did my master's degree. So this is the title, and we have a lot of authors from Africa and Asia. So I'm also here. I'm the second author. So this work was done on fall armyworm to know the genetic relationship of fall armyworm from Africa and Asia. So, and what we did, sample locations, the summary, accession number, and now before we use CO1, now we are using TPI gene. TPI gene is a nuclear gene. So CO1 is a mitochondrial gene. So we used both genes, both markers, CO1 and TPI, to carry out the analysis. And these are just statistical calculations. And you can see the bioinformatics analysis, which is the phylogenetic tree. This is just normal pictorial description. Now, this is the minimum spanning network. This is a population genetic studies that shows the relationship between the TPI from corn and the TPI gene from maize. I'll send the material to you can read more about why is it corn and maize. So you can see that we had this publication in is it MDPI? Yeah, MDPI insect. And the analysis, the bioinformatic analysis we did that is obvious here is just phylogenetic tree and this minimum spanning network. The others are just tables that are gotten calculation. So these are examples to show us that you don't need to do like sophisticated analysis before you have publications. So let me move on to the third part, which is much more complex than the ones we've been discussing right from the start. So this is my recent work also, just published. The work was done on mitochondrial genome. This is a full genome, about 15,000 base pair long, about the malaria vector Anopheles sinensis from South Korea. So now this is a short publication, and all you will see is just this phylogenetic tree. It's actually very funny. When I was just when the publication came out, I was just laughing that people that do not know what was done in this work would be like, is it not just this little work? But ladies and gentlemen, it's a more in-depth work that requires a lot of bioinformatics knowledge. So someone was want to stop. So this is also an example of the work that you can do from using bioinformatic analysis. So what is obvious here is just this small phylogenetic tree. But what you are seeing here is a combination of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Is about 15,000 times 10. That's around 1.5 million base pairs analysis. So this is just an example, but here it appears very simple and very little. So this is an example of works that can be done or analysis that can be done with your knowledge. And what we do here is just majorly annotation. We are trying to annotate the genes, and we've discovered where the genes are located. For example, between region 201 to 796, we were able to map that this region contains the CO1 gene. So that is an example of what we do in analysis like this. So, and there are lots more and lots more. Now, my latest publication in collaboration with the proposed speaker for today is was published in Nature Journal. So this was written about the need for more bioinformaticians for population studies in Africa. So this, as you can see, this little work is just about 200 word long. So it was published in a journal that is the impact factor is now 65, about 65 impact factor. So these are benefits, these are perks of being a bioinformatician and being a seasoned bioinformatician, that although it might look little to people that are viewing, so it is just but they are massive works that just infuse just simplicity. So now that is the first part of our discussion. So before I move on to the main topic of today, which is the career prospect, winning grants and scholarships. So do we have any question before we proceed?
Yes,
Any
I have a
questions?
question. Yeah, I have a
Yeah,
question.
please go ahead.
Yeah, my
And after
question is,
you, cheese, is it Mr. Cheese? Yes, after you, sir.
yeah, am I
Please
the
go
one?
ahead. Yes.
How does someone get to have a mentor? Like the processes on how to get a mentor.
And someone actually asked that also in the group chat. Getting a mentor. Okay. Well, by definition, a mentor is someone that has an experience in the field that you're about to go into. So we have different, like my specialization is in genomics. We have I have other colleagues that have specialization in transcriptomics and epigenetics and other subdivisions of bioinformatics. So getting a mentor should be, first of all, sliding into that DM, wishing to be their mentor. Or secondly, if there is a mentorship program going on in if there is a mentorship going on being set up by different agencies or different institutes or bioinformatics institutes, you can also explore the opportunity to be a mentee in that institute. For example, we made a call for mentorship and volunteering around February or March, and we concluded that program for 3 months. The program went on for 3 months, and it was concluded around uh, June. So we had like 6 mentees that we took through different aspects of the NOBLEKINMAT company as a whole, and also through the technologies regarding bioinformatics analysis. One of our mentees would also be presenting tomorrow, in person of Comfort Shekoni. So she's one of our mentees, and she's presently uh, in what's the name of this place in USA? So she's now a research, is it research assistant or research scholar or something? So, but she's presently in an institute in is he West Virginia? I've forgotten many things on my head. So she's presently there. So that's one of the perks. And then one of our another mentee of ours, who we also got admission to study in Germany. We also have another mentee who she's also into science communication, Sarah Nyachiri. She's a Kenyan. So we have a lot of, at least from our first mentee program, our volunteer program, we had success story. So we are planning to float another one soon, but the time and the commitments is kind of very tough presently. So that's why we don't want to come up half baked. But I can assure you that God willing, after this fourth cohort, we would come up with another mentee program. So although it will be much more robust than the first one. So yeah, about mentor. Next question,
Okay.
Mr. Cheese Cheese.
All right. Thank you, Mr. Presenter. I'm very grateful for the opportunity. You have
Yes,
opened
sir.
our eyes to so many things that we didn't know. But my first question is, can you publish a paper from the resources you get on NCBI or the Ensemble platform, the dry lab, just information, pick information from NCBI and publish a paper. If you have such a paper, can you show me an example of how it will look like and applying bioinformatic tools on such papers? And then the second question I want to ask is, how do I join the WhatsApp platform?
Thank you. Uh, for the first one, there are different papers. So, for ironically, I also made a promise to send it also, but I forgot. So I have not had that. I have not had the opportunity to publish with secondary data. They call it secondary data. So and in it is not that hard uh, to do that. So I've come across a lot of papers on that, but I will also share examples of that they use secondary data. It is very
I,
simple. Let me give you
I
an
would love to
idea.
see
Let me
one.
give you an
I would love
idea.
to see one. Yes.
Yes, yes, yes. Let me give you an idea of how secondary data papers come to effect. You might have, without even sweating for that, I have a publication I just read today. Where is it? Where is it? Where is it? Where are you? I just read the paper today. Yes, this is a typical example. It was published in Cell Press, iScience. I will share the link in the comments, or I download and share also. This paper was gotten with secondary data. So the paper, as you can see, it has a lot of authors from Nigeria. In fact, the authors are from Nigeria and Ghana, majorly from Nigeria. So a lot of the authors are from Nigeria. I think one from Ghana. So what they did was that epidemiology and genetic diversity of SARS-CoV-19 lineages circulating in Africa. What they did was very, very simple. They retrieved 5,299 complete genomes collected in 33 African countries from the GSAID database. We investigated the circulating diversity, reconstructed viral divergence and history, and studied the case and death of the country. It's very, very straightforward. What they did was that they carried out phylogenetic analysis, revealed that African viruses clusters more closely with those from Europe. So you can read more on this paper. I will send the link. Hopefully, okay, this is just a PDF link. I think it should, but you can search the title epidemiology and what's it called? So that's the link. You can check it out. So
Okay, I
this
really
is an
appreciate.
example, a typical example of using secondary data.
Okay.
So now let me give you an idea of how secondary data comes to place. This I will using this also as a platform. Uh, for example, there is this. I have this data for long. I have this data for long. I've been too lazy to come up with an idea about that. It's about Calosobrucus maculatus. I have like 12 samples. I have like 12 DNA sequences that are primary data. But I know using just 12, using just 12 is not enough. So to for publication in reasonable journal. So I've been thinking of how to, what I will now do is that I will just say the genetic diversity of Calosobrucus maculatus from Africa. So what I will now do is that I will go to NCBI. I will collect all the sequences that have been submitted on this particular insect of choice. I will submit it, I will download everything, and then I will do my analysis. Maybe phylogenetic analysis, any type of analysis I'll carry out on the work. I can do population genetic studies. I can do anything on that work. So that is one way. That those are ways in which you can come up with idea of uh, secondary using secondary data. So I think that answers the question. The second question is how do you join the WhatsApp group?
WhatsApp group, yes.
We have five groups already. So the stress of managing or posting and responding to questions on the five groups uh, was what led us to Telegram, because Telegram there's no Telegram channel. There is no limit to the number of groups. So now, unfortunately, also it was just a channel. You can it's a one-way channel, so it's not a two-way thing. So, but now that we have a group, we can also utilize that. But there are still spaces, but it's almost full in the full group. But now that WhatsApp has increased the capacity to 500, I think that would also contain a lot of people. So maybe we can create another WhatsApp group, and we'll share the link in the box. So thank
Thank you
you.
so
Next question, Mr. Efyong. Mr. Efyong. Okay, if there are no more questions, uh, good morning, ma.
Okay, thanks for the presentation. It was really clear. Please, I have two questions. Um,
Okay.
the how does one? I'm still on the and I'm intending to like have to do my project topic around bioinformatics. So how do I come up with a topic like around bioinformatics to like just a project topic that I could work on in bioinformatics? Then the second question is based on sequencing. Like you talked about collecting samples, and then you
Yeah.
run sequence analysis on there. In Nigeria, we have limited companies that do the whole sequencing stuff, like 54 gene and the rest of them. But I feel that at this point it's going to be really expensive. So how will a research student, like I'm still a student, I don't have like as much financial capacity like to be able to carry this out. So how do I go about doing a unique study? Though I just heard about the secondary research stuff, but maybe I could look into that direction. Please just help me out. Thank you.
Thank you so much for the question. So, well, what if I would ask, what field are you? Like, what field are you working on?
Okay, I mean, I'm biotechnology. I'm actually a biotechnology student.
Now,
Biotechnology.
please come again.
I'm actually a biotechnology student, and I want to like based on genomics, either genomics or proteomics in bioinformatics. But I'm a biotech student, biotechnology.
Oh. Oh, well, you have to pay for that consultation. Oh. So anyway, uh, it depends on your interest. I think a lot of people in biotechnology are now focusing on nanoparticles, but Nano is now outdated to me. It's now overrated also. Like it used to be the, oh, you are doing nano work. Wow, stuff like that. Back then when I was still in Footmina, because I started my master's in Footmina. So since then I've been like, why are people so freaked about nano technology also, when there are other technologies that they can focus on. So anyways, your work now, there are lots of biotechnological applications that are presently ongoing that you can use bioinformatics to explain. So so one of such is drug design and coming up with a candidate drug for a particular disease. That's that is a lucrative job field for you if you
Okay.
can venture into that as a biotechnologist.
Okay.
Um, in that way, since you said you are doing proteomics and the likes, so you need to do more of molecular docking, homology, and the likes. So gene ontology, and something similar to that. So that will give you the platform to launch yourself in the area of pharmaceuticals.
Okay.
So
Genome.
genomics,
Yes, that's my particular interest.
okay, that's your particular interest.
Yes. Yes.
It's easy. We have what we call GWAS, Genome Wide Association Studies. So if you have a need for yourself in that area, you can relate the topic, whatever you are doing regarding your biotechnology project. You can relate it to genome wide association studies to study the disease occurrence, the epidemiology, and the personalized medicine, especially for African populations. So you can explore. It's a very wide field that you can explore in full. So don't limit yourself. Just one thing about that is that you have to read wide. The reason why I'm lagging is because uh, I haven't. I want to explain in depth. Like, is that you know what you want to do. For example, I know what I want to do with my specialization. I am focusing on metagenomics and the bacteria that affect mosquitoes. I know that if I go deeper into my work, I can come up with a solution in which the mosquitoes will not be able to transmit the disease they are transmitting. They are transmitting. So there will not be need to eradicate the mosquitoes, one, which will be preventing extinction of organism. Secondly, there will not be a pollution of the atmosphere because the usage of pesticide and the likes, we'll be able to monitor that. So a lot of variety of options for me to explore. So what you want to do. The first thing you need to do is that you need to read wide. You need to read a lot in the area of that you want to concentrate upon, and then from there you can have an idea. This publication that we had in Nature was it was inspired by the second author in person of Abdullah Aborode, the speaker for today. So he just thought that, wow, since we finished our last class, our last session, actually the idea came out from our last session in NOBLEKINMAT of Bioinformatics, the third cohort. So after the sessions, a lot of people joined, but because there was not too much publicity for this one, so that's why there's a lot of reduction in the people joining. So he thought of the idea, why don't we write about this shortage of bioinformaticians in Africa? So from there, we can write a publication and let the world know that we need more bioinformaticians. So he just gave it to me as an assignment. Okay, write this one. I said, okay, my boss. I just wrote it and we submitted it and it got accepted. So when you put yourself in situations whereby ideas will come, ideas will come. So I think that's the best answer to that.
Okay. Thank you. The second question of like analysis in Nigeria. I don't know if you could
Oh,
like
yeah, yeah, yeah.
help us in sequencing our.
One way I think Nigerian scientists are doing it to cut the cost is the, for example, in this publication, I used forward and reverse primer. Even here in South Korea, is also not that very cheap, like cheap. It costs money. So here I used forward and reverse primers. So for each primer, they will charge you one. For each primer, they will charge you one same fee for two primer, and it is one sample. So one way I think researchers in Nigeria does it is that they will sequence just one direction, which is forward or reverse. So from there, they will now blast that sample with that of NCBI to correct whatever anomaly is present in their sample. So I think that is how they do it. That is one part. Second part is that you have no option. You have to spend money. So either you go and look for it, or you go and do GoFundMe for it. So you have to spend money. So there's no two way to that. So, but how to reduce the amount of
Thank you very much.
money you spend in Nigeria is that you also try to, like, I don't know the English to do that, not destroy the integrity of your work, but also to maximize the work you have. I know they do it in 54 Gene in Copa Biotech, African Biosciences, Nigerian Institute of Medical Research. I heard NITA is no more doing. They don't have the sequencer. So right now there is another popular technology that is very, very popular now, is the Oxford Nanopore. I think it was mentioned in our last classes, Oxford Nanopore. So what it does is the metagenomics and the transcriptomic stuff. So that one is cheaper, is a cheaper alternative to some of the sequencing ways in which they get the results. Although it is for another purpose. So, but however, there are cheaper options, and there are ways in which you can um, maneuver your ways. But in absolute terms, you need to spend money. So there is no way to that. God
Thank
will
you very much.
provide.
I hope
Hello, sir. I'm ready now, sir.
I hope you don't mind
Okay.
sending or listing the institutions you call that carry out sequencing.
Okay. Have I not been? If we have someone that has a full list of the company, please share.
At least the one
I only know, a few.
yeah, just this few. Like this, it could be.
Okay. I will share.
Thank you very
All
much.
right, sir. I'm ready, sir.
Okay, please go ahead, sir.
All right. Thank you very much. Though I have three questions, but I'll ask it one by one so I can get the response effectively. I'm also documenting, sir. Please, I want you to
Okay.
throw more light. I want you to throw more light on COX-1 gene. And why do you think, is it really better than other gene, or is it really specific? Because I know COX-1 gene has to do with mitochondrial DNA, and we also have other primers that target the genomic DNA. So why would you really prefer COX-1 gene, or what's the advantage of COX-1 gene over other genes that target other? I don't know if you can give justice to that, sir.
Very difficult question, but no problem. We would answer. So, one of the advantages of using COX-1 gene is that it is maternal. Now, usually, what we want to study is the evolutionary relationship, genetic diversity, and relationship between our samples. And the most prominent one, the most conserved, is the CO1 gene, because it is maternal. It has a maternal transmission. What do we mean with that? Between an insect, when an insect transfer the genetic material from the parent to the offspring, the maternal cytochrome oxidase is the one that is transferred from the mother to the child, from the mother to the offspring, not from the father. So that is why one of the ways in which COX-1 is very, very important. So its conservation, its stability, the size is also relative, and also it is conserved. Okay, I mentioned conserved. So COX-1, that due to its maternal transmission, that was why it is which is a choice marker to carry out evolutionary analysis. So that is what the most important importance of using COX-1 gene for population studies. Now there are other markers like the ND1. I'm sorry, ND2 rather. So there is also 16S. 16S, we have COX-1 and we have 16S. So 16S is a ribosomal DNA. We have the 12S, we have the 16S. Invariably, let me just put it in short. In the mitochondria, this work that's on display is a complete mitochondrial genome. So, and in a complete mitochondrial genome, there are 13 protein coding genes. There are 13 protein coding genes. Among the 13 protein coding genes, CO1 is the most explored because of the stability and because of the conserved nature. Now, once we know that this particular region is conserved, anytime we see a difference in the sequence of this particular region, we can easily differentiate between the samples easily, unlike other regions that are not conserved. So that is the reason why CO1 is used. The other one after CO1 is 16S. 16S is used a lot. So because it's the ribosomal DNA sequence that is but it is not part of the protein coding sequence we have 13. We have CO1, CO2, CO3, ND1, ND2, ND3, ND4, ND4D and ND6, ND5 and ND6, and is it up to 13 yet? So we have, let me go and copy. Okay, CO1, ND4, ND1. Okay, I mentioned ND4. Yes. So the ATP, ATP6 and ATP8. Those are the 13 protein coding genes in the mitochondria. So among all these 13 protein coding genes, so CO1 is the most utilized because of the conserved nature. So I think that answers that. Now, in other words, there are also other markers that are used. So in this particular publication, I mentioned that the fact that TPI gene was also used. TPI gene is present in the nucleus. So we can also use this particular gene for population studies, especially when we are dealing with we have insects or strains or organisms that has strains. Like, what do we mean by strains? They have the same genetic makeup, the same DNA, the same everything, the same makeup, but they are strains. Sometimes we call them cryptic species. I worked with cryptic species also in Bemisia tabaci, which is my first publication. They have the same genetic makeup, but they are different. They cannot mate together to produce a viable offspring. So these are some of the miseries that you would unfold when you go deeper into biological studies. So I think I was able to answer the question
Yes.
exhaustively.
In short, sir, I wish Nigeria would have bought one bottle of Coke for you to flex. You've really done justice to that question. I'm really impressed
You can
because
send it to. You can send it to.
all right, when I was researching on my study, I'm working on schizosomia hematodium, and one of the
Wow. Yeah.
gene
Yeah.
at the end, I want to use COX-1 gene, which will target the mitochondrial DNA, and I also intend to use another gene that will target another primer that will target the genomic DNA, which are ITS2 or DRA1 repeat gene. So, but I'm really interested in COX-1 gene. So I went into NCBI, I went to the old genome, and one of this thing I saw in COX-1 gene was, okay, I checked the genome, I checked the exon, the coding region. I noticed that it has 13 coding region. So one of the gene there was COX-1 gene. So I was so excited when I saw the COX-1 gene. I said, wow, this is what I'm looking for. So I drafted out the old gene. I downloaded it in a faster format, So I've been trying to say I can design a primer towards that. But sir, I want to find out this COX 1 gene, is it that we can also have COX 2, COX 13? Are there variants of this COX 1 gene in the genome?
So ITS2, the internal transcribed spacer. We have
Yes,
ITS1 and ITS2. I think is there ITS3.
yes,
It's
yes,
one of
yes,
the
yes.
most prominently used markers
Ah,
for fungi, especially.
ah, okay, okay.
Yeah, because it depends. You use a marker depending on the type of organism that you want to work with.
Okay, sir.
So, for schistosoma hematobium, yes, you can use uh CO1, but it will be preferred use 16S and
Um,
ITS2
16
because of
else.
the or ITS2. So because of their nature, 16 ITS2 is conserved in bacteria and fungi more than CO1. So that is one of the benefits. Now, moving back to your question, there are different variants of COX1 gene. We have COX1, we have COX2, we have COX3. So, but the most stable and the conserved one is the COX1 gene. So,
Okay.
to design your primer, it depends on what you want to work with. So you can design your ITS2 primer, you can design your 16S2 primer. But I will advise you not to waste your energy. You can simply go search online, search on publications that have worked on schistosoma hematobium, use their primer. There's no need to disturb yourself. It's now readily available. Like a lot of people who have done work on schistosoma hematobium. We can even help you search now, and don't let me say I'm 100% sure that you get results. We'll see answer. Let us check schistosoma, schistosoma hematobium, genetic diversity of schistosoma hematobium. Yeah, we have the results. So we would so in short, to save our time, you can search, and I'm very sure you'll see different information on that. So they use okay, this is RAPD primer. So and that's they used RAPD PCR primer, and they have the results. So we have the results. So you can try to uh we can try to do that and check that.
All right, sir.
So
Thank you very much. The second
you're welcome.
question, the second question is this, sir. Based on the experiences you recounted, how you kind of use a primer online, but you were passing through some stress getting the results. And sir, do you know that someone reliably informed me that there's some information you get online on materials and method, and when you try to replicate them, you can't get the result they got. And yeah, so it has been a challenge too, relying on information you see on journals. But I'm not saying that statement is absolute. Though. So what can you say about that, too, sir? Especially the
Well,
primer aspect, especially the primer aspect. You pick the primer, and or is there anything you need to do on the primer to work on it to confirm before you can go ahead? Or what advice can you give, sir?
for primers are they don't have problems regarding regarding if they work or not. So the only thing you would have to change, if it is the same species, you don't have any problem. I have not had the cost to design a primer for myself. I have not. I have not had the cost to design primers for myself. I use readily made primers that has been designed for the work I want to do. So I do not have problem with that. The only problem I have is doing the analysis itself. So not with the PCR part. So um well, I think that's I have not personally I've not had the
Sir,
course
please,
to.
can I add something to it, sir?
Yes, please go ahead.
Okay, I understand what he's saying. There are times that you buy primers online and then you work and it doesn't give you the same results. I think that's
Oh,
I think that what he's saying now refers back to initially when you started the class that you need a mentor. I had a similar
oh
problem when I
yeah.
was doing my work, when I was doing my own work, and the primers I bought didn't work. But my supervisor, who is a mentor to me, when he came in, we started looking at the PCR conditions. Most times it's not that the primer is not working, but it's about the PCR conditions. So just a little alteration in the PCR condition from what you see from the journals was what really
Thank
bailed me
you.
out. I got a perfect
Thank
result.
thank you. That answers your question. What you just described, I also had the same problem too. So it answers the question perfectly. I didn't get where he was trying to go, because I also ordered some primers. I followed the same protocol. I didn't get the same result. So what I did was that I just tweaked the primer conditions, and then voila, we had the result. So that's it, Mr. Effian.
All right, thank you
Thank you
so
so
much for
much.
the last screenshot. It has been so insightful. I'm grateful. The last question, sir, so that others can handle the floor too. You know, one of the problem, or will I say the disease caused by schizosoma hematobium is bladder cancer. And I know that there are other factors that are
Bladder
implicated.
cancer?
Yes, it causes bladder cancer. Yeah, schizosoma hematobium, it causes
Or
bladder cancer.
it's pathological
Implicated.
manifestation leads to cancer.
Okay, okay, yeah, yeah,
Complication,
yeah.
complications.
I think you're getting it well. Maybe I'm not using the right words, sir.
The right word is
Yes.
complication.
Okay, please say
Yes.
it again so that I can learn. Say it again.
Complications
The
of
path
schizosoma hematobium is bladder cancer.
I didn't get it, sir.
I said complications of schizosoma hematobium is bladder cancer, not that it
Oh, oh,
causes, it's a
oh, oh,
complication.
oh, oh, thank you. Please let me take note of this. One of the complications of bladder of schizosoma hematobium is bladder cancer. Thank you for that correction, sir.
Yes.
So now my question is this. There is a research I want to embark upon. I know we were supposed to do docking, and I would need that knowledge and experience, sir. I want to a kind of isolate or recognize a kind of target protein that is implicated in this bladder cancer. Then use drug extract and plant extract and a kind of generate a ligand and carry out docking aspect. So I don't know if anyone could throw more light on how I'm going to embark on this project. It's something I want to embark on, please. I need a sound guide on this, sir.
come on, please.
Pay knowledge is power. Pay.
Come on, please.
Oh, these are things that did come by.
It will take us a lot of time actually to explain. So,
Even if
but
it is a kind of idea, sir, because I want to embark on it, please. I need a guide.
well, we can speak on camera and behind the camera. I
But
actually
sir,
don't have an idea
Hello,
presently.
good morning.
So,
Sorry.
please come in. Good morning.
Mr. Efron,
yes, sir.
I would
Yes, ma.
advise you are going more than a question. You need to do consultation, so you need to pay a fee. So you should meet him privately. Seriously, it's
Thank you.
beyond free. Thank you. So we can proceed to the other things we have. Thank you very much.
Thank you.
All
Thank
right,
you for
thank you,
that,
thank
for
you,
saving
thank
me.
you.
Next question, please. Okay. If we don't have a question, I think
Okay,
we
okay.
can
I have a
proceed.
comment.
Okay, sir.
Yeah. Thank you very much. Yeah, even though Mr. Effiong has really made my day, and also your comment, and this brother Keola has also helped my day. I really appreciate that I'm here, but I also want to let you know that I'm also working on co-hygiene for my masters. We are still on it. I'm working on genetic diversity of Xenopus muleri and Tacharina pumilio using co-hygiene. Actually, those two species are edible frogs, that we can get in major parts of Africa. Understand, and we've decided to choose cohygiene. So when you were answering the question that okay, cohygenes are very conserved, I was in the same, because we had to do literature review and why are we choosing cohygiene. But I learned more because you started talking about the 13 protein coding genes in mitochondria. I don't know that, but now I know that it's not only I know that it's not only cohygiene that is a genetic marker that can be used, but I think I have learned more about some other markers. We are still sampling. In fact, I'm looking for people that can help me get some of those edible frogs. We are working in group. So, but it's 3 frog species we are looking at: Oplobatruca occipitalis, known as African bullfrog. The Xenopus muleri is a molars claw frog, and then the other one also. So, but we are still looking for the sample, because without the sample, we cannot get our DNA extraction and then proceed. Understand? But I'm optimistic that if we are able to get the sample across the 4 ecological zones in Southwest. We are actually looking at Southwest. Nigeria is too big for us now. We don't have money yet. So, but we are looking at the southwestern part of Nigeria, and in that part we have four ecological zones. We have the mangrove swamp, we have the rainforest, we have the derived savanna, that is the rainforest gradually already getting desertified, and then the Guinea savanna, that is the southern part of the Guinea savanna from the north. And it's like, for example, the Shaki, the northern part of Oyo State, fall in that area. So we are looking at 3 locations in each of these ecological zones, and then in each of the location we are to get the samples of the three species of frog. But my supervisor actually gave me 2 of it, so Xenopus muleri and Tachana pumilio. We are on it, but I'm actually how will I put it now? I'm actually excited about the work, even though I'm still kind of new. But I know that once one keeps learning, one keeps rubbing mind with people of like we have on this page, one will start getting the picture. And you said something that really helped me this morning. I hope I'm going to be rounding up soon. Is that mentorship? Some of these are not easy just by reading books. You almost go crazy. I'm like, what are we doing here? But the more we have someone to guide us, the more we are willing to learn, the more we are willing to unlearn, then we begin to get a clearer picture. But I would love to get funds for this. If there's anybody that can help me or put me through on how I can get for, so that I can really enjoy the result from this. Imagine we have to get those frogs from those areas. It will really be a very good thing for conservation of frogs and then molecular work. Thank you very much for allowing me to share my thoughts.
Thank you so
Sir,
much.
please, I want to know the last speaker's name so I can connect with him, please. I don't know his name.
Is it me or Mr. Akeola?
No, the last person that just spoke, the last speaker.
Okay,
You.
my name is
That's it.
Sunday Eludodun.
Okay, Sunday. Okay, I'll search on you.
No problem. I'm on LinkedIn.
All right.
You can search for Sunday
Okay.
Eludodun, but yeah, you can.
All right. Thank you,
Thank
sir.
you. Yeah.
All right, thank you so much for the contributions. I wouldn't want us to take much of our time, so let us move on to the next sections, and then we can have more contributions and questions after that. So, by the next point is the career prospects, the winning grants and scholarship. So, I will be practical with this part, which is I would say explain from my own personal experience regarding how I was able to get the scholarship. So one thing is, not everybody. I want us to believe that, understand that not everybody
Thank
would have the opportunity to have scholarship. but everybody has the opportunity to progress in their career or how to win grant. So the first part is getting a scholarship. So I actually wrote a book on how to write a statement of problem and study plans for scholarship application. But this is there in no way an advertisement. I mean, if it is an advertisement, that's good. It's good for market also. So, but anyways, so one of the ways in which you can get a scholarship is through forums like this. I will identify like three, three places in which you can access information regarding scholarship. One is joining a forum of like minds or people aiming for the same goal as you. Through that place, you would have access to a lot of information regarding scholarships. So, secondly, I also do share sometimes scholarship opportunities regarding bioinformatics. So, it is not also limited to bioinformatics alone. It also involves other area of endeavor, which includes other aspect of specializations. So you can also be part of that. So join yourself with groups of like minds. Secondly, know how to use LinkedIn. LinkedIn is one of the most potent tools in which you can have access to information and know more about your field. Connect with people that have already excelled in that area of study that you want to achieve proficiency in. So the third one is know how to use Twitter. The simple hashtag on Twitter can lead you to any information. I want you to put it into practice. After this class, I want you to search hashtag bioinformatics, then hashtag bioinformatics scholarships, hashtag bioinformatics scholarship internship. So just you can change the search features as much as you want. So I'm very sure that you have tons and tons of information regarding your next scholarship regarding bioinformatics. So these are examples of ways in which you can get access to information to scholarships on bioinformatics. So the fourth part is that you join. Okay, I've mentioned groups, but groups on WhatsApp. There are lots of groups on WhatsApp and Telegram. Telegram has made it so simple now that you can search for scholarship on Telegram, and it will give you different options. It works like Google. So you can search and it will give you different answers to what you search for. So those are areas in which you can search for scholarship. Now to my own personal story. I was recommended by a mentor here in South Korea to come here for studies. So that's how one of the ways in which you get scholarship is through recommendation. Nowadays it is very easier but competitive to get scholarship because one of the ways you can search for scholarship is searching online. For example, the publications I just shared now. If you are interested in the work that the publishers did, the emails of the corresponding author is always there. You can write a letter of intent to the author, telling the author that you are interested to work in his laboratory, that you saw his work on so, and that you have started the work on so, and this is what you want to produce, or this is what you want to proceed further in. So that is one of the ways in which to get that. One of my seniors is now he would graduate in next, I think this month. One of my seniors, he wrote letters like that to about 600 professors. Out of that 600 professors, only two responded. Out of the two, he eventually got one, and now he is now a PhD graduate. He started PA Masters and completed his PhD under that same professor. So If you do not have that dexterity, that commitment to search, to work, to labor, you are on your own regarding searching for scholarship in that aspect. The second way to search for scholarship is through this popular scholarship program. We have Chevening, we have Erasmus, but not everybody have that qualification or opportunity to get selected from, because they are neck-breaking competitive, very, very competitive. So the first one I explained to you is one of the easiest way to get admissions into places that they offer scholarship. So that is that about scholarship. I would have loved to go more, but time. I want us to finish this class before, uh, because I have to also attend some other lectures. So then, moving on to winning grants. How do you win grant? I was lucky to anchor a program in with Professor Musa Abiodun Aibinu, the VC of the Vice Chancellor of Summit University. Now, thank God Allah is here. So the Vice Chancellor of Summit University 2 weeks ago, and he discussed in details how to access scholarship. Mr. Abdullah, please, you can share your screen. We have been waiting for you. I've been holding the fort for you. So, I was the moderator for that event, and he discussed about how to access grants, especially if you are an academic in Nigeria or any other African countries or any other place in the world. So I'm going to share that PPT material with also in the Telegram page, and I'm very sure that it will be very, very helpful. It is very, very useful to every one of us. So I wouldn't want to go on into the details on how to get grants, but I know there are other, that material will serve as enough guide to put us through. Then the career prospects. I want to briefly go about that because our facilitator is here already. So on the career prospects regarding bioinformatics and any other aspect, especially let us move on to bioinformatics, is it's very lucrative. Bioinformatics is a very lucrative career presently. The baseline salary, especially in the US or anywhere, is that you are paid about $80,000 per year, depending on how you are able to negotiate your uh, deal of contract. $80,000, at least $80,000 a year. My other colleague, in person of Dr. Austin, is a senior bioinformatics consultant, and my oga is doing well. He's doing well. So, and one good thing about being a bioinformatician is that you can work remotely. You don't have to be where the work is. You don't have to go to the workplace before you do your work. Just take the works and then do it remotely and then give them the results. You can have like 3 to 4 jobs, and each paying full-time salary for you to you. So that's one of the perks. It's like working as a tech guy, and we know that tech is the new oil now. So you can also be a beneficiary of that if you get to that point in your career. In your career, and one of the extra things that you can do to be a prominent bioinformatician with high recognition and financial gratification is that you add not just the bioinformatics skills. There are some other skills that are attached to bioinformatics that if you have that skills, you'll be an hot cake. So one of that skill is the statistical skill. So if you have the data analytics skill, it will be a lot of benefit for you. We mentioned that in one of our sessions. You should know how to program or code, maybe R or Python or what other programming language? The Python, R. We have Java. Any programming language, especially the R and Python are the most popular one, but they are not the most most valuable. there are some codes that are used to write software, bioinformatics software programs. So you can use those codes to also, you can, if you are good in programming, you have a space in bioinformatics. So another aspect is database management. Mr. Abdullah, please, you can come in anytime you are ready. So another aspect is database management. One of the skills is, for example, the popular one, Amazon Web Service. If you are proficient in Amazon Web Service, we also have Alibaba. We also have Microsoft Web. They have uh, different web services, database management. So if you are proficient in that, ladies and gentlemen, you are good to go in bioinformatician. You'll be eating from different aspects. Like money will be, you will not run broke. You will not get ever. You will never get broke doing that. So these are some of the ways in which you can engage yourself with. Another aspect is data science and machine learning. Machine learning is one of the ways in which, as a bioinformatician, it will be of great benefit for you. So, my Oga and I, Mr. Abdullah, we recently published a paper on machine learning, using machine learning in drug design and other aspects. I will also share that the file with us. So, machine learning also has application in bioinformatics. So, it's a wide variety. It's a buffet. Just pick from anywhere you want to come from. So that's a quick summary. So I will give the floor to Mr. Abdullah. Please unmute yourself. Oh, I have seen you. So I think
Good.
now the floor is yours.
Um,
Thank you so much for listening. The real original owner of today is here. So he will take us through
uh,
this. So he will
uh,
take us through the journey.
yeah, good morning or good afternoon. We are early in the morning here,
Be- Before,
5:20.
before, before you begin, sir, I'm leaving the floor to you. Anytime you wish, the break, anything, the floor is yours now. So Mr.
No, no,
Effiong is
no.
here. Mr. Effiong is there.
No, there is no problem. There is no problem. Yeah.
Alright,
Yeah.
sir.
Good morning, everyone. I really appreciate the invitation. Um, and I'm glad that everybody is here. Um, the aspect we're going to treat today is on career prospect, scholarship, and grant writing. There are three different things entirely that need three different aspects or different area. But I'm going to start with career prospect because that is the basic foundation or the fundamental for uh, grant writing and scholarship because it need experience. Now, every field has different aspect, and each of the field have what you need to do to gain experience. If you want to develop yourself in a career or in an aspect, there are things that are needed. Part of it is number one, you need the skills. Number two, you need a an interest. What what is your interest? That is number two. Number three is you need to work. That is number three. Number four is you need to have an achievement, an accomplishment of these two things that you mentioned. So I will start. And as an example now, as a chemist, if you want to develop yourself in a career, the first thing to look at is, as a chemist, what is the area that I am good at? That is the first thing. So how do you determine the area you're good at? You determine the area you're good at based on how you understand the concept of that area. Now, there are branches in chemistry. there are branches in microbiology, there are branches in biochemistry. Now, each of these branches have what it entails to become an expert in this branch. You can't wake up suddenly and become an expert. It's not possible. But you can train yourself, learn and get trained to get experience. Then the more you now practicalize the skills that you acquired, the more it tends to grow. Growing is the prospect in the career you decided to choose. And each of the career has target. What are the target? What do you want to become with your career? Do you want to become a professor? Do you want to become an emeritus? Do you want to become a scientist? Do you want to become a research scientist? Do you want to become an industrial scientist? Do you want to become anything? You have to map out what do you want to become. Where exactly are you going with your career? There are some that their career are being achieved based on their career are being achieved based on what the aspect of the level at which they are looking at. Oh, this is the apex level that I'm looking at. An example, you want to do a bioinformatic, you want to become a bioinformatician, or you want to become a computational chemist, or you want to become a computational chemist, or you want to become a structural biologist. This is a career path. Then how do you want to become it? That is the prospect, that is the growth. Prosperity in the career deals with growth, and growth comes with rigor. What I mean by rigor is work. If you don't work, if you don't put much hard work to the training and to the skills that you acquired, the career will be stagnant. It won't grow. And another thing entirely that attached to the career and interest that determine if you are growing in your career or you are not growing in your career is, are you truly learning? That is number one. Number two, what is the result of what you are learning? Did people in your field truly know you, who you truly are in that field? Are you truly contributing positively, greatly to the path that you decide to choose? That is why I do tell people that the first thing to do, once you know that this is my interest, okay, I wish to work on chemical biology, that is my interest. And in this chemical biology, I wish to work on protein or DNA or nanoparticles to find solution to treat, you know, communicable disease or non-communicable disease. Now, I've decided on that. What did I have? What did I have as a tax that can push me to acquire this? The first thing is that, did I have sufficient knowledge to push the career forward? If I have BSc and I aim that I want to become a professor in bioinformatics, I want to become a professor in chemical biology, and I don't do what it takes, I don't pass through the rigor to become that professor, the career expansion will not go forward because I need knowledge to grow, to reach that apex level. So what did I need to do? I need to go and do, if I have BSc, I need to go and do master, or I need to go and do PhD. Okay, if I acquire that, that is just a general theoretical aspect. Did I have the skills that will let me or allow me to be able to think out of the box? Then I will need to acquire the skills that are needed. Did I have the instrumental skills? Can I set up a lab myself? If I don't have, where can I learn it? Who are those people teaching? Who are those teaching people about this aspect? If it is computational based, did I have computational skills? I wish to become a bioinformatician, but I hate computer. So if you know that, that means going through bioinformatics career will not be a directional prospect for you to build your career on. Because if you want to, whatever, wherever you want to build your career on, must be an aspect that must be able, or that you must be able to contribute perfectly and proficiently. At the same time, you must be able to retrain and get people trained in that field. And all involve skills and knowledge. So, and also interest. The more you have interest in a particular area, the more you get more, the more you get more acclimatized with that field. And the more you get acclimatized with that field, the more you get comfortable in the field. And the more you get convenient and comfortable in the field, the more you'll be able to reason widely and be able to create a new gap, a new knowledge, and answer new novelty in the field. So if you don't have interest, but you wish to have interest, you need to learn. Maybe when you are learning, at the part of learning, this is the aspect that will build your career, that will push your career into another level. And what is the level of building your career? It will build your career in the form that people in the field will truly know who are you. They will know that Dr. Ashraf is good in computational and bioinformatician, and is a good bioinformatician that have did or that have conduct or that have explored several researches. Yeah, that have explored several researches in reputable journals. Because why I'm laying the emphasis of this is that the career prospect is the build and foundation for your scholarship winning, for your any accomplishment that you want to accomplish in academics or outside academics. So, and that involves a lot of skills, a lot of knowledge, and a lot of interest. The passion you have, personal passion. Yeah, personal passion. What you decide that it has become personalized with you because it is your career that you are building. So, as I said, building career involve knowledge acquiring, skills acquiring, and interest. So once you now have interest, this is my interest. This is aspect that I want to work on. I really love this aspect. I always have different idea about this aspect. I wish to explore this aspect. Then I have the rigor knowledge that I need. I have the basic knowledge. I have the integrative knowledge. I have the applied knowledge. Then I've learned from reputable uh, scholars in this field. I've learned different skills from them. I've explored these skills. I've explored it. I've seen the result of the skills I learned, and I have, either published it or have integrated it to the real world, and it's working. Then what next? What next is to work to produce all what you have learned. This can come in form of laboratory, can come in form of going to academics, it can come in form of going to industry. Now, there are now different words in career building when it comes to these three things. Either going into academics or going into industry or going into real world. You want to do an entrepreneurship or anything. Now, basically, I know that most of people that are here are most of academicians, so I'll just touch academics and industry. Building your career in academics is different from building your career in industry. They are two different worlds entirely. But the most interesting thing in building your career in industry, as I've said, building your career in academics is based on maybe two or three things. Number one is based on what are the quality of things you are producing. Yeah. You can have 100 papers, you can have 1 million papers in academics, but what is the quality? Yeah. And what is the prospect of what you're doing? Like, what is the benefit of what you're doing? Did what you are doing add into the knowledge gap of the field? Can other researcher in your field, you know, reproduce what you are doing in form of citation and create a new gap or a new answer to the question that have been brooding a lot of scientists in your field? So that is why when you want to build your career in a high path, an high way, that you get a lot of recognition in scientific world, you must do some research that are of great quality. Because if you do research that are of great quality, it will be publishable in highly impacted journal. And if it is publishable in highly impacted journal, you have autometric. The autometric is you the visibility at which a lot of people around the world will know you will be so high. So if you're answering a lot of answers in that your field or interest that you select, a lot of scientists will reach you out, either for collaboration or for better explanation to the entanglement that they have of part of what you have answered, which might be a leading way for them. With that, you have built most of your career to the Apex Act. That is number one. Then number two is that creation of lab. Yeah, you can have your own lab yourself, dealing with a specific area of feature or menace in the society in form of disease, in form of anything. That is number two. Then number three is that you your salary level will be so high when they see your output. And in academics, the university that you are attached to, other university might feel like you can come as a visiting researcher, visiting scientist. They might even collaborate with you that you'll be working in your school at the same time working in several universities as a visiting scientist. So these are part of recognition, and you'll be able to meet people that have high caliber in your field, learn from them, collaborate from them with them rather. At the same time, network with them, because what really matters a lot in academics is network. The more you have network, the more it's easy your part in the journey of your career in academics. That is about academic career. The academic career, it's all about developmental of knowledge. At the same time, producing of tenure papers, quality papers in reputable journals. Because if you want to build your career, you have to publish in Scopus Index journal of reputable journals that are aligned to your research field. So once they align to your research field, people in your field will recognize who you are. Who is Mr. Abdullah? What does Mr. Abdullah did or does? If I want to work on that part, I know who in my field that did or that are doing what I'm looking for. And with that, I'll be able to go a long way in my journey of academic career in terms of recognition, in terms of being well-known scientist, and in terms of solving problems in the society, which will lead you to the path, or which will lead us to the path of scholarship and winning grant. Then, before I go there, I'll go through industry. Yeah, industry is a very wide area that is another different world on its own, which is completely different from academics. Yeah, completely different from academics, because you need to redirect yourself. Yeah, you might be good academically, but there are two ways. Yeah, I know some professors or scientists that explore both academics and industry. Why and how? It's very easy. If what you are doing are novel, another question that will come in is, are they scalable? What I mean by scalability is are they something that they can be explored, or they can be redirect, or they can redesign in industry? Can industry implement what you discover so far? If that is the case, if industry can implement what you discover so far, that means it's scalable. So once it's scalable, the industry can remodify what you have discovered in a form that public can relate, public can go with it, public, it can solve public problems. And what is that? Big money. Because the industry, you have to pump more money into your research to discover more. And before you know it, your lab will grow as a big lab. That is one of the apex level of being a career researcher. You grow as a big lab, you have about 40 students, 20 PhD, five postdoc, 3 visiting lecturers, you have supportive other scientists that help you throughout your laboratory. So, and once your lab get money, a lot of people want to join your lab. And the more a lot of intellectual people is joining your lab, the more your career is expanded. Because when you're talking, oh, I have about 20 PhD students that I've trained, that have graduated, they are now in industry, they are now in academics, they are assistant professors, they are doing well, executive, they are scientists, researchers in industry, and they are doing well, collecting huge amount of money. So when you are talking, people in your field will give you that accolade and that recognition. That means you have reached an apex level where your career is recognizable. But as I said earlier, one of the first things that deals with career building and growth, so that you will not be building on a path that doesn't have a foundation, because no matter how high you build it, it will fall one day. If you build your career based on fake perspective, fake interest, dogmatism, because people are doing it, you want to do it, you want to build your career based on uh, based on based on fake knowledge. Yeah, because there's fake knowledge. There are people that doesn't go to learn about their career itself before they start building on that career. An example, we've seen a lot of young people nowadays. I mean, I have some people that I know that I stopped working with because I found out that literally they don't know about research. They don't know about research, but they want to do research. What they need to do, as you that want to build your career on research, what you need to go and learn. There are a lot of coursework online that are free that you can learn about research. There are a lot of conferences that talk about research training. There are a lot of group of people that teach people how to do research. There are a lot of mentors that that are publishing in highly impacted journal. This kind of people can teach me on how to do research. Learn from them, be their mentee, stick with them, keep working with them. Research is not something that you learn in a whole day. It's a process. Same to a career. Career is a process. You shouldn't jump much in your career when there is difference between jumping more and dreaming high. Dreaming big is good. Everybody dream big. Everybody want to become. I want to publish in Nature. I want to publish in Science. I want to publish in immunology. I want to publish in big journals. But did I have what it takes? Is it my baby review, or my the lab that I do? I'm doing some research that in the lab I know that this kind of research cannot be published here. So I need to step up my game. How do you step up your game? By improving your skills, improving your knowledge. So jumping in the research, it involves like what you need to do and you don't do it. When you reach the apex level of your career and it get to a point where you need to defend yourself, you find yourself that you are not fitable to that aspect of life, of where life have taken you to, and it will become a shame. Because when people in your field start finding fault in what you're finding out, and they keep asking you questions based on what you find out, and you cannot give answer because you don't have idea. or knowledge about it, then you have failed. People will not recognize you are publishing several journals, but what is on your brain? What do you have? Are you braining? Can you be trainable? Can you train others based on what you are writing? Can others be like, oh, I really appreciate this man because of him I'm being refined in science, and today I can call myself a whole scientist, a whole, not half. They bake you fully because they are fully loaded. So that is the aspect of building a career. If you are building your career, you build other people's career based on your growth. And the more you do that, the more you have experiences. So you cannot give what you don't have. That shows that you cannot grow, you cannot grow based on what you cannot grow in a career that you don't have anything in. You don't have, you are not well equipped based on skills and knowledge. The rigor is not there. So it is a design that need, a journey that it's step by step. That is what I would just say. It's one step at a time, one step at a time, one step at a time. Before you know it, it all compounding. It compounding. People will need the skills that you have. When they need the skills that you have, you apply for it. They take you. You keep building your career around it, and before you know it, before a whole year complete, you just know that, oh, I can say these are my achievement for this year. I've grown in this career. I've known a lot of people in this career. In this field, they know me. In this field, I know people. So basically in industry is completely different. I've seen a lot of scientists that are doing they call it integrative research, cutting edge research that can be integrated into real world problem, that can solve real world problem. People are organic, they synthesize catalysis. Catalysis are being helpful to solve some, some challenges or some reactions that result into real world problem in industry. People are redesigning, structures to help or to ease the researchers or scientists in industry based on what they're discovering. People design chemical reactions, reagents for industry. People people design a solution, a target. They rediscover protein, they rediscover protein targets that can solve a likely solution in a real world problem and that can be integrative in industry. People also find answers, several answers to a lot of problems that can help industry to find a redesign or a product out of the solution, a product out of the solution. Yes. So basically, I can name like someone, like a prof. Her name is Professor Shalini Prasad. She is from UTD. She is doing a cutting edge research that is integrative, that based on sweat. She she works on sweat to redesign a target that can solve cancer, other tumor disease and non-communicable disease through the target will be used as a model to design maybe an app or equipment, biomedical equipment that can be useful for industry as a product to sell for people. And firstly, did she not have a big lab? She have a lot of grant. She have industry collaboration. A lot of industries collaborate with her with a lot of funding every year. So now, whenever our students are graduating, most of the industry take them in because they are doing what they are doing. So most of them have they have already get to a point in life where they give solution, they design solution that are useful for these industries. So now I cannot be an industry. Now I've seen people that are solving problems that pertain to my industry. I'll take them in. Why? Because they are resourceful. They are resourceful will boost my industry. They have experiences because that is what they have did or they have done for years, like 5 years, some even 6 years. So their productivity will boost my company. And once my company is growing, I get a lot of profit. I'm part of the profit, put it back into the research to boost more solution that can that can that can alleviate my industry in industrial world. So these are another career path chances that are okay if you are good in what you're doing and you are doing a cutting edge research. I've seen some African researchers that discover anti-malaria drugs, antifungal, antibacterial, anti anti-inflammatory through metal ion. But the question is, are these research scalable in industry? Because when you get to industry, they have to see the scalability to the real world. It has to be a product, it has to be a new design, which involve new skills. That is why many scientists or many researchers do learn about entrepreneurship, MBA. Know how to do business? Can you do business? That is another career path on its own. If you know that your research is cutting edge and you truly want to integrate it to the real world and get a lot of money, you want to find a solution to antibiotics, or you want to discover a target that can be used in form of drug as to reduce the influence or impact or effect of antibiotics. They discover a drug that can be useful for the whole world. Then you said, okay, we are the one that will be producing this drug. We collaborate with the pharmaceutical company to do this work. Everybody has their own cottage or profit that will be collecting. The industry will have theirs, you will have yours. Then how do you collaborate? How do you integrate your business idea to industry so that industry will be able to collaborate with you? It's another different part on its own, which you have to learn through your skills and knowledge part. You have to learn that. If you know that where I'm going, I will one day I will find a novel thing that will be useful to the world. And if I want to collaborate, how did I collaborate with the industry? See, when I become a professor, when I don't have much time, is when I will start go and learn about business. So I have to integrate all the aspects in the learning, so that when time comes, it will so easy for you that when you are designing a solution that you are designing, you'll be building a business model with it. So in industry, typically, not to waste time, in industry are there are many to involve, pharmaceutical industry, computational industry, gene industry, research industry. So it all depends on the prospect that you are looking at. Oh, I wish to work in farm companies. I wish to work in research companies. I wish to work in computer companies. If you want to do that, do you have the skills of as a starter? Okay, you want to work as a research assistant in a research industry. Do you have idea about research? Are you good in that company research, like the company research, what the company is doing or what the industry is doing? Are you conversant with it? Are you good with it? Are you convenient with it? What level of experience do you have? The experience is a time of learning, failing, and success. Learning, failing, and success. You learn, you relearn, you unlearn, you fail, you succeed. So, what is the level of your experiences? Have you conducted research in that field before? Can you thrive if they give you one aspect in that industry to work on? Because if you have level of experiences, you have level of rigor of knowledge, you you are conversant with everything in that field. You can do research in that field. You can work in the laboratory because you are a whole scientist. You are complete. When you go to industry, it will be so easier for you. But at the same time, for starting, you will need to learn what are the rudiments in that industry, because each of the industry has their own relearning and unlearning. What you learn in academics might be completely different from what you'll be doing in industry. So you have to unlearn you with some of the things that you are bringing. You have to integrate some of the things that you're also bringing into what you are relearning. So ability to perfectly and greatly integrate part of what you have learned so far to what you are relearning would make you a brilliant scientist in industry. And before you know it, you can have your own industry and make millions of money every year. So I speak in a general terms because we have different people in the feed, and I hope it will be kind of quite helpful in this part. So this is the part, or this is the part of career prospect that I would love to stop here, then go to uh, grant winning. Yeah, that is the most interesting part because I've already break down what is really needful or what you need to know. Grant winning are different form. You know, we have field grant, academic grant, and industry grant, government grant, and non-governmental grant. Yeah, government grant, non-government grant, that is NGO grant. We have uh, academic grant, industrial grant, and feed grant. I will explain each of these one after the other. Feed grant can be in form of international grant, like international organization like UNICEF, UN, WHO. You know, there are many. So there are so many international organizations. But why do I call it feed grant? You know, I call it feed grant because it is based on you going to the field to note out problems and create a solution to that problem and make a report. That is about field grant. It's always sponsored, or they are always sponsored by international organization. You go you go to the field. What I mean by feed is the target. The one target, like about 18 to 35 years old people in Nigeria are living in poverty. Create a proposal that can solve or can relieve the poverty level that among the youth in Nigeria. Everybody will send their proposal. Oh, Mr. A has a proposal, Mr. B have a proposal, Mr. C have a proposal, Mr. D has a proposal, Mr. F have a proposal. Now, what is the best proposal that stated the research problem? What is the source of poverty among youth that justify the problem of youth, that results to poverty, that justify, objective that can redirect and recreate and, pointed out solutions, policy recommendations for the youth. So you now go to the field to go and meet the youth themselves, which is your target population. So find out what is resulting to poverty among them. Then what is the way forward? So the fund, international organization fund huge. Or another example is on menstrual hygiene, on, they can say you should do research on menstrual hygiene, do research on manners in your society, do research on civil engagement and democracy, do research on different aspects. It depends on what the international organization stands for and what they really need as a report on the whole year. Do report on education in conflict affected area. The fund Can you find a solution to those children that have of school? How can they go back to school? These are the reports that they modify and publish for policymaker to see, so that when they are making policy, they can use part of your findings of you that have go to the field. You have seen the children that are being affected. You have lived with them. You have talked with them. You have relate with them. You have engaged with them. You know their problem. You know the source of their problem. You know the state of their problem. You know the future prospect of their problem. Then you can modify a policy and a solution that will have, integrative measure, like stepwise method to solve their problem. So winning such a grant involve you to have speedy experience when it comes to volunteering and leadership engagement. You may be able to, depending on your field, like an example, what I used to tell people is that you have to engage with SDG. When you engage with SDG rather, Sustainable Development Goals, each of these goals has indicators. They have, goal 1, which is no poverty, goal 2, goal 3, quality health, goal 4, quality education, goal 4, quality education, goal 5, all like that. Each goals have each indicator. Now I'm interested in goal 3 and goal 4. That means that if any international organization is calling out for grants on menstrual hygiene, I can work on that. If they are calling on non-communicable disease like cardiovascular disease, what are they are calling out for grant on that, I can work on that because that is my interest. That is my SDG interest. I know much about it. I've been into the field. I've seen more sufficient. So I can be able to create something out of the problem that I've seen to the theme that the international organization is looking for. So if I can do that, they will be able to relate with where I'm coming from to what they really need. And with that, you have a very unique and fantastic proposal. But I will talk about how to modify a proposal, either a fit proposal. I will talk about that. An example now. I have someone that was one of my friend. He is one university in Europe, and he is entitled as a scholar to find a problem either in his own country or in the current country that he is. He should find a problem that is affecting the public and create a proposal that will have solution to that problem, that is solvable to that problem. Then after that, you'll be if your proposal is quite well, you will win the grant. Then the problem that the man want to select is on SDG Go 3. He reached me out. He was like, "Oh, you are doing a lot on SDG Go 3. What is the problem that you think that we can write on, either in this own country or in Nigeria?" I was like, "Okay, let's talk about Nigeria. Oh, there's problem on part, the level of poverty. They call it part poverty. The level of poverty that comes with woman buying part, with the level of inflation that comes with part, is so high that so many girls cannot afford to use pad every month. The money they will use to use pad or to buy pad, we quite quench the level of hunger for some weeks. So why can't we create a reusable pad that every girls can reuse for, they can reuse, and at the same time we come in a level that they can afford. So ability to do that, you have solved one of the big problems that is affecting the society. So we design a problem, we make it a research interventional based project. That is interventional bridge. We did the project ourselves and do research on what we have learned so far, which is we create an empirical study. So our personal view and perspective of what we have learned the experiences we have had. So they do not only give us the whole grant, they give us extra grant when they see the report. So in feed grant or international grant, when you do beautiful work based on the grant that they give you, the next cycle of grant that is coming, you might collect more than what you collect earlier. I've seen one of my friend uh, that is working on nicotine. It all start from 2019. Yes, he won the grant in 2019. He is working on nicotine. So it's all about harm reduction. Yes. So instead of people to be smoking, so why can't we have nicotine that will reduce the harm instead of smoking cigarette? So he do research on Nigeria smokers and those that are not smoking. So he was able to win the grant. Then the following year, with the level of his result, he won another grant that is higher than the first one that he won. Then today he won a grant that worth $60,000 in that field. And he's just a graduate student, he just graduated. So basically, it's all about going to the field. When you're able to go to the field, you'll be able to know where the problem lies. This is the problem where this is the problem most governmental people they are not getting right. They are not talking to the people that are in the field, or the people that are in the field are not giving the real feedback to the government people. This is where the problem lies. This is a policy that you need to formulate to redirect this kind of problem. Or they are not even going to the field. They are just developing their own view and perspective, which might not work in the real world and real field. So ability to solve a proposal, a field proposal is all about your experience and ability to think a problem and a solution. That can win you a field proposal. So, and note this is very important in a field proposal and winning a grant in field. You have to have experience. Because it is part of the rigor they are going to look. What is your experience wise? Even it might not be many, but have you do enough that can make us sure that you have the knowledge and experience in this field? So this is how to win international grant. A lot of international grant, feminism movement, LGBTQ community, water, water problems, water scarcity, developmental entrepreneurship, a lot that you can win. It all depends on your field. What field are you working at? Climate problem. Yes, it's there. So it all depends on what field are you working at, what is your interest, how do you want to push, forward with this interest. That can actually solve your field problem and field proposal. So your field proposal is all about problem solving. Then your academic proposal. Your academic proposal is all about research. Yeah, that is it. It's all about research. And to be able to win academic proposal, there are levels. There are early career researcher proposal, there are mid-career researcher, there are professional one, which is for professors and emeritus, So early career researchers, it's highly very competitive. Why? Because you have to have a new novel idea that are relatable, not only in your field, but can bring about multidisciplinary research. That is what most professors do look like. Can your research create a multidisciplinary approach? If it can create a multidisciplinary approach and it can also solve problems, then you are doing something that are very great, that need to be boosted on. But sometimes your research may only focus on your field, but is it creating a new knowledge? What is the new knowledge are you creating? That is one thing. Is one thing to have a new knowledge. That is one thing to have a new knowledge. Is another thing the way you establish your point. the way you present your point, the way you write your lexical structure and your English determine. There are some people that they find it hard to just, they know what they are doing, but they find it hard to establish it in word. Like people can see, they can relate with the the intuitive they have based on the research. So if you want to write a grant as an early career researcher, you must be able to have, you know, you must be able to have a new knowledge gap, or problem solving research. That is number one. Number two is that you must be able to, have a good structure that will establish why you are writing this. Why, what, how, how do we need to solve this research? Why are you solving this research? Why is this research important? Then one of the things that also, will be very helpful for early career researcher in grant winning is having a mentor. Have a mentor in your field that can help you to proofread everything that you have in your proposal. You should have a mentor, a correct mentor, and you must be able to ready to learn, and must be able to you must be able to must be somebody that uh must be teachable. Yes, yeah, you must be teachable. There are some why whereby you have fantastic research, and your mentor might redirect you and might refocus you and might be like, oh, I will do what is inside me. I might not follow my mentor comment or something. Yeah, it do comes like that. I'm telling you, mentor is, it is something that, that is very important in the field, that you must have somebody that you are looking into, somebody that will train you and get you trained. And having a mentor is very, very easy. As an early career researcher, your mentor should be a professor in that field. People that have won grants so many times. So they know what is really needful in your field, because each field have different things that are needed. If I'm writing on public health now, an example, I'm writing a proposal on public health now. One of the things that attract more public health research is that, are you doing a research that is qualitative or quantitative? That is the first question. Is it an original research, or is it a systemic research, that is systematic review? That is number one. Then what are you solving? What is the problem? That is number two. Have people written about this problem? That is number three. If people have written about this problem, what are you bringing that is different? And that is number four. So these are the questions that they will try to look in your proposal. So if they don't find out that gap, they will consider your proposal as you just write English and you just send it to them. So quite all well. Career grant are always competitive. Early career grant are always competitive. But what make it more interesting and award winning, that you're able to win, is what is the problem you are solving? Can this problem's solution, can the solution of this problem bring differences in the field? Yeah. So can other people lay more knowledge on this answer? Is it multidisciplinary? So that is it. So for middle career researcher, you have been in the field for years. You have had a lot of experience. It's about, some people do write their research based on what they are currently doing. Yeah. For mid-career researchers, assistant professors, doctors, they write research based on what they are currently doing. What are you currently doing? Okay, I've been working on Staphylococcus aureus for years. I've I've had about 15 papers in the American Microbiology Society (AMS). I've have my paper in Nature, in Elsevier, in Springer, and they are good and fantastic research. You will list it even as an early career researcher. They have publication in the field you're trying to work on. So what we allow you if your research is quite unique, what they allow them not to, have much pressure on them, is that they are still coming, they are learning. So how do you now want to see that you have the skills to solve that problem? Is what you present in your CV and your cover letter. So which lab are you working? That is the question. They will ask you, which lab are you working? Because the name of the lab that you are working also counts in your grant. If it is lab that have all the needful thing that you said you would do, that have it, or the one that doesn't have it, you can't be doing a research that involve NMR, IR, that involve a lot of big instrument, and you are in Nigeria, you are in Nigeria laboratory. The question is, how do you want to do it? So you must be able to know, and you must be able to know and reason before you write a proposal that you don't have the skills and available equipment ready in your hand. So you must be able to have that. Is very, very, very important. Very, very, very important. So as a mid-career researcher, one of the things they'll look is at what is your field experience and what is your track record. Have you win grant before? You have not win it before. Do you have publication in that field? The field that you're writing at. Yeah. What is the track record? How do you publish? Where do you publish? What is the quality of what you publish? They will check everything. Who are your mentor? Who have you mentored in the field? So, and what are you producing in your lab? So these are the things for early career researcher to look at. Your mid-career researcher, their own grant is based on what they are currently doing. What are you currently doing? So then the professor, they have been years in the lab, they have used years. They are using grant to solve new problems. Yes, a lot of new problems that they have that they feel like it need to be solved. They have huge number of grant before. They have huge number of students, but they feel like this problem is something that need to be solved. They can write grant based on that, and they can get it. It's not a must that they have to get it, but most wise they always get it. Professors and emeritus, because the the level at which they write and the level at which the art of writing will be so simplified that everybody in the field or anyone that's reviewing the paper will know that this is an art. Because when professor writes, you know it's an art. So they write clearly and at the same time in a technical form about their idea and what they need to solve in their lab because they have everything. So another type of grant, is governmental grant. I don't know if that is mostly common in developing country, but in developed country, government gives a lot of grant. So the government I mean is not a federal government. Federal government has their own grant. State government has their own grant. Ministries have their own grant. Ministry of Army, Arm and Defense, Ministry of Environment, Ministry of Education, Ministry of this and that. They have yearly based grant that they give out to people that are working in the field. An example now in US, you see NSF, you see NIH, which are most common. NSF and NIH is very, very common grant that most professors do win in science. NIH is when you're working on, health research. It might even be bioinformatics as far as it's solving health problem through scientific approach. NSF also is basically for science, anything science, you get, but it's very, very competitive. Very, very, very competitive. So what would make a difference is about the type of proposal you are submitting, and I will still lay emphasis on it. Is your proposal, is it something that someone has done before, or is it the first time someone will do something like that? That is why when you are doing when you are conducting a research that won a grant, you have a lot of trial and error because it is something unique. It is something no one has done before. So you have to be keeping trying, keep failing, keep trying, keep failing, keep finding solution until you reach that area that you are going. So governmental research grant is very, very common. And at the same time, it helps a lot of scientists in the developed world because that is what they live on. And that is what most of their lab is being based on. So And then we have non-governmental grant. This is for NGOs, and those that have NGOs are into leadership and into making change. Yeah, change makers, for change makers, for leadership, for leaders, and for those that are making quite impactful and resourceful things in the society. So if you have NGOs or if you don't have NGO, but as far as you are making impact, you can apply for NGO grant to support your your journey. If you have something that you are doing that you want to support on, but you don't have money, you want to give out to people, or you want to do a project that can change the life of people, or the life of an example, you want to make a project that can take children that are out of school back. You need money. So you can apply for NGO grant. There are a lot of NGOs that are giving grant. If it is education, we have Malala, we have UK Aid in Nigeria, we have Global Education Forum. We have a lot of NGO that give a lot of grant for people that are trying to solve educational problem. That is for NGO grant, and that is all about NGO in a while. That is all about grant winning. So the last part, which is the last but not the least for today, is you know is scholarship. Yeah. So scholarship is a very wide thing. As I said, it's very wide, and it's very comprehensive. At the same time, it's very simple and tactical. Yeah, that's what I would say. Very tactical in nature, because at the same time, it involves prayer and hard work and resourceful accomplishment. Yes, ability to show what you have done. So, because people that are reviewing your application doesn't know you, there are a lot of scholarship, academic scholarship, industrial scholarship, you know, uh, academic scholarship, you know, research scholarship, industrial scholarship. So it can come in form of different aspects, but one of the most common one, which we are currently, that are into academics, you love to venture into academic scholarship. So we have different type of scholarship. We have undergraduate scholarship, graduate scholarship in form of master or PhD or postdoc, visiting lecturer, blah blah like that. So one of the most common one is that I mentioned is master, PhD, and postdoc. There are three most common scholarship, and each of them has different ways at which they look at. But we are looking at academic scholarship, which is either undergraduate, master, PhD, or postdoc. I'll start from postdoc. You know, Getting a postdoc scholarship involves years of experience. That is number one. Number two is that involves you being able to showcase an accomplishment throughout the years in the field. That is number two. Number three is that ability to be able to work in the lab you're going to. So now there are a lot of advertisement that people might say, oh, they need people in the lab. You might send your CV and you might not hear anything from them. So one of the problem that I do say is that, okay, now I'm working on bioinformatics now, strictly bioinformatics and computational and genomic research sometimes. So that's what I'm working on. But I want to do my postdoc in, you know, computational or, bioinformatics research. What I need to search for is professors that are into bioinformatics. Now, when I search for professors that are into bioinformatics, I have to look if they are looking for postdoc. Yeah. Some people might just be mailing prof that doesn't even need professor. That doesn't need uh that doesn't need student. Yes. So I know there are some that need student that quite hard because they will not reply you. Yes. So one of the things that I do recommend for postdoc is that number one, you have to have your CV with a lot of your accomplishments. State everything that state all your strength. Never feel like you want to limit yourself by just reducing your CV. State everything you have achieved. They want to see everything you have achieved. State all your strength. State where you're good at. State all your publication. State everything. state your Google Scholar link, everything, state it. Then you have to have a cover letter about your interest in that research field. A cover letter that states your interest and that states why you are motivated, and if you have been you know you are being allowed to work in the lab, what you will be able to achieve. You have to state it. Then if you have to use either an institutional email or an email that involves full of your name, full name. So you have to have that. At the same time, you have to have, if you use some institutional email, sometimes it give more, you know, more more kind of reputation. But if you have a full name that at a Gmail, full name at a Gmail. Then another thing entirely is that if you mail prof and the prof didn't reply, one of the things that you can do is that you search for a student, either a past student or current student in that lab, and mail them. Either you can search them on LinkedIn, you can search them on, find a way to contact them and ask. So professors are always busy sometimes. So if you're able to go through their student, you may be able to reach the professor. That is one of the trick for postdoc, you know. So I'm visiting researcher. So one of the game is that you will see a lot of advertisement, keep applying. There will be one that will ask you for interview and blah blah like that. Then you know that you tend to talk to people that are doing postdoc currently. If they pass through interview, or people that can train you based on interview, how to ask question and how to answer question. So now for master and PhD, depends on country, but I'll just state the general things that it is needed or needful for you as for you pursuing master or PhD. Number one is your CV. Number two is your SOP. Number three is your personal statement. Number four is your recommendation letter. Who are your recommender? Number five is your standardized test. You have either GRE or TOEFL or IELTS or Duolingo. Number six is your publication or any research experiences. Number four is do you have teaching experiences also? Do you have working experiences? Then all these things are very, very necessary. Now, there are a lot of mistakes that people make when it comes to writing CV and SOP, which I will just touch a little bit. Then I will just take the criteria of acclimatizing the step at which you can apply to different scholarship in schools or in graduate schools and the likes like that. Then I will round it off. So your CV should be your strength. That is one of the things that there are a lot of people that there are some scholarship that have their 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 guide about how their CV will be, their format. You get, but still in that format, you have to state your strength. You have to state your strength because they don't know you, they don't know who you are, they don't even know where you are coming from. Some people they will go and ask anyone that come from similar country to ask about, do you know someone from, do you know this university? Hope this university is good. I have someone coming from this university, or I have someone I'm hoping that I might choose or I might select to come from this university. Do you know? Like, can I take it? Then I take them in. So, but ability to see what you have achieved, and like, wow, that is great. This is what I'm looking for. Because every lab or every school or every department has or have what they are quite looking for. Like, what are you looking for? What is your strength? Your strength really matters. So if you have your strength, you won't have problem. You know, so you won't have problem. Your strength really matters. So you have to showcase your strength uh, in all your CV. And what is your strength? Your work experience, your teaching experience if you have one, your research experience. both undergraduate, graduate, if you do master or if you have master, or if you have PhD also. Then your certifications. You have certificates based on the course you are going to. And one of the things that people mix is that you have to have certification that align to your research interest. Don't go and have certification that are not in alignment to your research interest or your experience. Yeah. So if you are working on drug discovery, and then you are adding certification on social civil engagement, and in your on your CV we don't have any volunteering work that you do. So it is not aligned. So add certification that strictly align to your experience and what you have already established. The another thing entirely is, do you have publications, your research outlet, or the one that you are currently writing, or the one that are under review, anything? Do you have it? So if you have it, you have to add it. Which year do you publish it? You put the link, the DOI, and then you can add the impact factor also if you want to add it. That is not a problem. Then another thing entirely is, what are the skills? What is your laboratory skills? What is your technical skills? What is your soft skills? You know, you need to state that. What are the instruments that you can operate? What are these and that you can do? And you have to state that. Then another thing entirely in your CV that really matters, that really, really matters is your leadership experience. Have you led before? Are you a leader? You know, you have to state that. It's very important. So before you know it, you have a substantial, enough CV that can push you and that can portray whatever you are writing in your SOP. What is needed in your SOP, statement of purpose, is your background. Why or what does your background bring you to, or inspire you, or motivate you to do to this course? Like, what bring you from scratch to be motivated to study the course you're currently engaging with? Like, okay, now what make you study chemistry? That should be, tell a story. That is the first paragraph. Tell a story. What make you become, or what make you make, or what make you, you know, what make you, or what will make you to study chemistry? What motivates you? What inspire you? Then number two is that, what do you do during your undergraduate when you're studying chemistry? There you have to state your academic excellencies. If you don't have, or if you are weak at that, you have to first state your strength. You state your research experience, you state your teaching experience in form of compounding one. You have to state it in a level at which you'll be able to understand. At least two paragraphs we state about your research experience, how you feel, you know, how you feel, and how you, when you fail, how do you come back from your failure? So then you have to state um your teaching experience also, if you have one. Who do you work with? What are the professors you work with in your lab? And how it is easier for you. You get. Then you have to state your accomplishment with all this. Did they give you an award? Do you win any award? Do you publish any of your paper? If you publish any paper, you have to state that I will publish paper on this and this and this and that. Then the next thing is that you have to state your experiences, your laboratory skills. Let me say skills generally. You have to state your skills. What are the skills that you have? Then you have to state why the university is one of the best for you. You know. Then after that, you have to link it to why or how that university, or how the faculty researchers that have research interests aligned to yours. Then you now state your short term and your career goal. That should be everything in your SOP. So if you want to have a good and you know you know engaging SOP, and your SOP, what you're most stating, your research interest and your publication must align to what is on your CV. Everything must align. That must not be different. It must not be different. So then your personal statement is about the personal experience of yours. You can state about your personal experience in the lab, like When you're growing, you are inspired and motivated to become this and this. So you face these social challenges, you're able to beat these social challenges. Then you are currently working as a research assistant in this, you then expatiate more what you cannot expatiate in your SOP. You expatiate it more in your personal statement. So personal statement is as if you are selling yourself, who you are, you know, this and this and that, I've do this and this, that are completely different from your SOP, like more of like buttressing point. Then you can have your research statement. If you have one, you can have a research statement that state your research interest. Just a one page document. Then you can have your standardized test if you want to do, or if your scholarship actually require for it, GRE, TOEFL, IELTS, and Duolingo. So they are research interest to standardized test. So you have to take one that you wish. And if you feel like you don't need it, so it's okay. It's not a problem. As far as you have experience and you have skills. So and your recommender, they are very important. They are what makes your application complete. You have to know who your recommender is. Are they people that that can actually push you, and then they can push you at the same time. They can uh you know they can they can push you to do more, and at the same time they can help you when you fail. They can inspire you and motivate you, when you fail, and at the same time they can you know I remember something. uh they are ready, they are mobile. You can reach them anytime. Yeah. Sir, I need your recommendation on this sort of paper. Pa, 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 pa. They have did it. So they are mobile because one of the important aspects of your application is recommendation. So with this, I hope that you have learned one or two or three or four things about career prospect, grant winning, and scholarship hunt. Yeah, I wish to stop by now. So if you have any question, let me know, and I will answer you correctly. Thank you.
Wow, 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 wow. How will I put it? I'm saturated already. The chemical, the chemical. I've seen a lot of people who have questions. I've seen a lot of hands up. Mr. Effiong, Miss Adebi Dokaza, I think it's Dr. Patricia. I'm not sure. I think it's Doctor. Yes. Okay, you can go first before Miss Dorcas or Dr. Dorcas. The title I didn't on.
Thank you, Mr. Blake. Like it was very impactful. First of all, the first thing I was finding the career prospect. So for, I'm not observing this challenge. I'm not a pure science person. I studied education, biology. So and I have a research interest in biology. I have been trying to navigate from education to biology. I've taken some to cancer and a lot of stuff. But I'm still looking at myself as adequate for the career transition because first of all, I don't have enough experience. So how do I go about it? And number two, when you talked about this scholarship and every other thing. My question is that someone like me now with research experience, and I write the professor that aligns with my research lab experience. I don't can I reach out to the professor?
All right. Thank you very much for that. I really appreciate your question. The first thing is about navigating and transiting into your research. and your career in microbiology. Am I right?
Yes, yes.
Microbiology and what?
Research and cancer research.
Cancer research. All right, great. So now there are two different things entirely. Yeah, I get in microbiology research. Microbiology is a whole branch of a whole branch of a field. Like is a field on its own. Are you getting me? There are lots under microbiology. You have clinical microbiology, chemical microbiology, you have plant microbiology. So it a whole field on its own. It's a whole field of study. Are you getting me? Cancer biology, it is specific field. Are you getting me? It is a specific field of interest that people do research on. That is, they do research on biology of cancer. And they're finding a target or solving cancer treatment through biology techniques. Are you getting me? But microbiology is a field on its own. Are you getting me? So if you want to transit from microbiology to cancer, from microbiology to cancer biology, is very cool. But one of the things that is, as I said in your career path, is that knowledge, skills. Do you have the knowledge that can make you transit to cancer biology? If no, have you acquired the knowledge yet? That is the question. So if you will be able to acquire the knowledge that can allow you to transit to cancer biology, that will actually ease your path when you get to cancer biology, because you'll be able to understand the rudiment and the situation of cancer biology in the world right now. What is the current research in cancer biology? What has been the yesterday research about it? What is the new development of tomorrow? But if you don't have the knowledge and the skills that from the field that you are coming from to where you're transiting to, it will be very hard for you, because the level of productivity in that career will be low because you'll be struggling. So, except if you are a biochemist now, it's always easier for biochemists you know, to explore cancer biology, or anybody that have had similar experience, or that have worked in maybe cancer lab before, and yeah, they have already had similar experience. It will be quite easier for them because they will have read and have knowledge about cancer biology. Because cancer biology is very, very wide, and it all depends on where you want to venture, what techniques you want to venture when you are dealing with your cancer biology. So that is the first one. So if you want to port, or you want to explore cancer biology, you have to, you can think of how can microbiologists translate to cancer biology. What are microbiologists doing on cancer biology? And you can look at professors that have microbiology, that are using microbiology techniques on cancer biology field, in cancer biology field rather. So that is another way. That is the two way. Is it that you go and get more knowledge and skills that pertaining to where you want to venture into cancer biology, or explore what a microbiologist can do when it comes to cancer biology research? That is the first question. Then the second question is on when you don't have research experience. Yeah, that is why you need to do intern. You have to do internship. You have to have research experience before you go to big lab, because it will be very, very overwhelming. Even though when you get to big lab, they will have to reteach you again. They will unlearn you and you'll relearn. Are you getting me? But when you have some knowledge, you have modern knowledge in you, it will be quite easier for you when you are relearning. Are you getting me? So it is best for you to go and learn about research and work as an intern with some big industry that are doing research, like NIIMR, and other industry that pertain to microbiologists, NAFDAC and all like that. So it will be easier for you to learn some equipment, play with all this equipment, you know, uh, how to work with that equipment also very matters. So at least it's quite helpful. So I hope I answer your question.
Yeah, thank you.
Mm.
Please, please, I want to contribute to the question she has.
Yeah, you go ahead.
Okay,
Go ahead.
so I think where microbiologists and
cancer biology can merge is looking at the role of some organisms such as bacteria that act as probiotics in cancer treatment and prevention. That's the way. So just search and you see a lot of work that has been done on that. So that's just my own contribution.
Thank you very much, ma. Yeah, thank you.
Dr. Patricia.
Yeah.
Now you wanted to ask. You had one that asked last. Um, Mrs. Dorcas or Dr. Dorcas.
For the opportunity, sir. Thank you, it's Ms. Dorcas. Thank you so much for organizing this session. It was so impactful. I've been really blessed, and thanks to our guests also for the enlightenment. So I don't know if I'm supposed to ask the question now because it can be so interconnected, but I think I can ask on mentorship and internship. I know you mentioned some labs while answering the previous question, but I was not able to really get them specifically, where we could have internship to have the research experience mentioned. And also my question will be more like a personal question that I wanted to ask initially based on career path. So I don't know if we have platform for direct mentorship, especially for someone like professors that you mentioned in academics, and also probably someone that is in the industry, that is top there in the industry, that we can connect with. Thank you, sir.
All right. The first question is on. Sorry, pardon. I only take that of mentorship. Sorry, please, pardon for the first question. Okay, I remember now. Internship and in places to work at work. Yeah. Okay. Yeah. If you are in Nigeria, there are list of companies and list of organizations that you can work with. But the first thing is that where does your field align? So now, if you are a chemist or you're a microbiologist or you are a biochemist, there are a lot of organization, federal based and private owned based organization. So I know it's quite hard to see internship in this organization because of the level of network that you need, before they can quite employ you. But I know of some federal based on institutions that are quite free, that all what you just need to, you have to pay for your training. Are you getting me? And you learn. They won't pay you. Are you getting me? But the problem that we're having about most of the undergraduate students is that most of them want them to pay them during their internship. And that is a problem for career. So you that want to build your career, you don't go after money. You have to first learn. You have to learn. You have to acquire knowledge. You get from experiences of people that hire. Then later money will come. You don't have to run after money. They'll be using money to beg you
Should come and
because you have the skills.
back too.
Yeah, you have the skills. You have the skills, and the world need the skills. And exactly, you have the skills. The world need the skills, and you are here. They will be begging you. But when you are after money, what you're supposed to learn, you will learn it, and you will be half baked. So I know of NAFDAC. I know of there is one industry, FIR something in Oshodi. I know of NAFDAC is two. For people that do economics and food science and the likes like that, they can work in NAFDAC in Oshodi. Even chemists can work there, biochemists can work there also. They are almost doing the same analysis. But if you want to work with drugs, you can go to CDCL in Yaba, Central Drug Control Laboratory in Yaba. I think for NAFDAC then it's about 7,000 or something if you want to work as an intern. So, but for NIMR, National Institute of Medical Research, also in Yaba, it's not far from NAFDAC also. That's CDCL. Yeah. So what is it?
Naima.
Do they have to?
NIMR, yeah, sure. So it's, I think they pay based on your level. Are you undergraduate? Are you graduate? So I don't know much, but for undergraduate, I think they pay. I don't know. I don't really. I'm not well sure, sha. So, but you have to pay some certain amount of money as an undergraduate, and as a graduate, you have to pay certain amount of money. I know people that doesn't have they don't have experience in research, in lab research, but when they get their scholarship, they get with their past experiences. Are you getting me? They have to go there for like maybe 2 months before they travel to learn, and they pay them. So you can explore different lab there. So and then having that curiosity, be ready to learn more. Your curiosity must be there. Like, be ready to learn more and be ready to ask questions from people. And nothing about getting to know about mentorship. Yeah, Dr. Ashraf is there. I'm also there. If you need anything inside the group, you can message us. And if you need a mentor that are in the feed of your interest and you want to align to them, you can actually connect you to them or give you their reachable numbers that you can reach them and ask them any question at their convenient time, and to be able to help you. So that's it.
I think someone has already also added to the information regarding the internship. They said 25,000 for undergraduate and 60,000 for graduate. 3 months for undergraduate and 6 months for graduate. What I'm not getting is, are you the one to pay, or are they the one?
Yes, you have to pay.
Okay, okay, okay.
You have to
So
pay.
that's
Yeah, that is for NIMA.
Sorry, let me
Nigerian
Yeah, that is for
medical
NIMA. Yes.
research.
Yeah.
sorry. Let me add to it. NEMA fee has increased actually. It's now because it's now around 35 for undergraduates now. They've increased their payments.
Wow.
Okay. But
So
I'm telling you, what you are going to learn will be mind blowing than your mate. I'm telling you, because they have a lot of section in NIMA. They have a lot of section. And if you are a curious someone, you are very curious, you know, you can explore all the lab before you are done with your program. And you going back to school, you are different from your mate. So you have been baked with some aspect of life in your feed, and which will make it different. So you can go ahead.
Okay, thank you so much for your responses, particularly on NIMA. I've been to NIMA before now. I'm running my master's program now. So I was there before, and knowing that I'll have to make the payment, the question in my mind wasn't about the money, but it was about after this, then what?
After
So I believe that yes, after the training, then what? So I get the certificate from NIMA, and I believe that one of the top places where I can like say I'm starting my career is like NIMA. So after this, where am I taking this? They said I'll be giving letter of recommendation or something to show that I passed through that place. So where am I taking it to in Nigeria? That was like the problem I had then. But with this exposure that I have now, so I know that after Naimade, I'm going to
exactly,
a better place. So I could apply to a better place, probably outside the
exactly,
country and all
exactly.
that.
Yeah.
So thank you
Yes,
so much
yes,
for your
yes.
responses, sir.
And at the same
Another question.
Right. At the same time, you can add more to, you can build your career in Nigeria. You see, the problem is about you identifying a problem. Are you getting me? You can build your career from Nigeria and you'll be well recognized. We have many good scientists, here in Nigeria that are doing well, they're doing fantastic work. And they have quality, they produce quality things that I know. An example now, I have a lab in one of my friends is inside that lab is on environmental research, and I show him some scientists from Nigeria that publish on environment, and was like, wow, we are quite working on this. This guy have already published everything. And the guy is from Nigeria. You know, they are working on voucher of plastic waste, and the guy have almost typically published most things on voucher, most things on waste and plastic waste also. Was like, wow, if he was saying that if my professor lay my hand on this guy, you know. So you can build your career from Nigeria. It's all about, have you identified any problem you want to solve? This is always the problem that most scientists face. You know, have you identified problem I want to solve? I want to start working on HIV, you know. That's what I want to start working on. I want them to recognize me from Nigeria. And do you know something? You can rechange the story. So going abroad, starting from Nigeria is almost the same. So when you start from Nigeria, you have created a name for yourself that when you get there, your name will still start growing more because people already know you in the field. And anybody that is working with you, they know that you are quite resourceful. So that is it. Yeah. Okay, any question?
Yes, a
I have
question
question.
that you did not talk about undergraduate scholarship tips.
All right. Undergraduate scholarship is it's kind of very comprehensive a little bit, but I'll just discuss about the little I know. As far as I'm concerned, there are a lot of requirements that are required for undergraduate, but one of it is your SSC, the amount of A that you have or the amount of B that you have. Because strictly A or B do it. Now, after that, you have to go for SAT or GMAT. It's very important. If you want to win a scholarship, you have to have SAT or GMAT. Then you have to have TOEFL. Or sometimes people do A level. Are you getting me? You have TOEFL or IELTS, or you do A level. If you have all these, the next question is you have SOP also, like a graduate. You have CV also, and among one of the really thing that can't, if you are not going to US, and are going to other universities, your volunteering experience, your leadership experience. It's not must you have publication. Your volunteering and your leadership experience really count a lot. What have you volunteered for, and what have you lead, and what do you hope that when you get your undergraduate in that school, are you going back to your own country to do better? You get, but in US you will need SAT or GMAT and TOEFL or IELTS, and then you try to apply to those schools. There are schools that not all schools offer scholarship, but there are a lot of schools that offer scholarship in US for undergraduate. Yeah, I hope I answer your question.
Yeah, Mrs. or Dr. Kemi Olalekon, I discovered that you normally do not know the caliber of people you are speaking to here, so I think I will adopt the doctor.
I don't
And
adopt the doctor. I'm a mister. Thank you so much, sir. We appreciate
you're
the,
welcome.
the presentation and the whole program. It has been highly rewarding. I want to ask the presenter just few questions. You mentioned about mailing the professor for those that are doing postdoc, that we can probably reach the professor through their students. Is that also applicable for graduate studies? Is that also
All
applicable
right.
for graduate studies? Then also, if you have to mail the professor for graduate studies directly, what should be the content? Maybe I didn't hear it or network took me away. I didn't get that. Then lastly,
All
what
right.
I want to ask lastly is
Okay.
this. I'm someone that um, I have at least a lot of experience in things that have to do with molecular biology, genetics, but I don't have my master's, and yet I don't have my PhD, obviously, if you don't have master's. So I'm thinking of if it's possible if I can get a PhD directly, rather than splitting the two programs. If I can get something that can give me everything as a one program, and I also have some publications that I've worked on. So I need your advice, sir. Thank you, sir.
Yeah, that is fantastic. That is really amazing. For your first question is a million student. The major reason why I said talk about million students is because most of the professors are very extremely busy. They have a lot of tasks, and not all the time that professors will check their email. Are you getting me? So once you're able to know their for postdoc, are you getting me? Once you're able to mail their student, you know, if their student reply you, they will be able to get in touch with the prof for you, and the prof might get back to you if you truly need student or you have get a student. Are you getting me? That is based on the decision, because postdoc is on decision of the professor. Are you getting me? It's the professor that will decide that he truly want a student, not the school or not the department. They will be the one that I'm recruiting a new postdoc and it's coming to the department. You will tell the department, and the department will do all the normal formalities. Are you getting me? But when it comes to mailing professor for graduate student, yeah, you mail professor is good. Yeah, reason why is that this is what I do. If anybody give me a school now, they give me a university that, oh, check this also university, University of Southern Mississippi. Okay, this is the university. Before I even write anything pertaining to that university or submit any application, I do two things. Number one is that I search their faculty. Did they have my research interest? That's number one. Did they have my research interest? When I start by faculty and I say like, okay, there's some research in this department I can do. I fall in love with one of the professor or two, at least two. Get if one didn't want to have one, we are always accept you. One or two, at least two. You have at least two professors that you really want to work with that have research interest based on your background and your experience. And say, okay, yes, you know, they have it. Because these are the mistakes many people make. They went to go ahead to apply to a school, and eventually, when they get to that school, they went to go and do research that are not pertaining, or they don't have idea of, or it's very hard for them to crack. And they become frustrated. They will be overwhelmed. Yes, because you'll be overwhelmed in graduate school. Very overwhelming. Get so now when you go and do a research that you don't even have idea of, or you just want to be pushing it from the start of your graduate school to when you, if God just give you that energy, you may be able to see it through, but you will not be happy for one day. Yeah, because it's choking. So you want to do the research, you already know that. Then number two is to mail the graduate coordinator of that department. Their process, if you want to ask a question now in the graduate coordinator, before I mail a professor, you know, I've already noted the professor, I've named them their name. I'll now mail the graduate coordinator. Dear graduate coordinator, my name is Jesus so. I'm a graduate of this also with this also. If you have a very low GP, it's not a must for you to include your GP. But I am a graduate of this also university with this also grade, or graduated with first class, or graduated with second class, lower or upper or third class with this also university. If you do GRE, you can do your GRE. If you do TOEFL, you can do your TOEFL. If you do Duolingo, you can. If you don't have it, just state what you have. I want to apply to this school and I want to make an inquiry for this for 2023. So below are my questions. Number one, can I apply with my profile? You know, that's number one. Number two, you can attach your CV. If you don't want to attach your CV, just state some of your strength in the first statement. Can I apply? If you don't want to apply, can I apply? Number two, do you waive standardized test? Like, okay, you can do it like this. Do you waive GRE? Do you waive TOEFL because I'm from English speaking country? Do you give funding? Because that is the important thing. Do you give funding in form of assistance or research assistant? Number five, do you waive application fee, or do you give application fee? Application waiver rather. So once you're able to ask that, he will now answer you. Oh, you can apply. Our deadline is this. Oh, we give teaching assistant or RA. Oh, we waive TOEFL or we don't waive TOEFL. We waive GRE or we don't waive GRE. Oh, we do this and that. He will tell you. So you are now going to look at what do you have, what you don't have. Do you have all in his answer, or you don't have all in his answer? Are you getting me? So if you have all in his answer, now you jump back to the professor. Are you getting me? That is when you mail the professor. You get what I'm saying? So now, if you don't have all the answer, you don't mail the professor when you don't have the answer from the graduate coordinator. Then when you now mail the professor, this is how to mail the professor. Dear Professor something, You have a, a subject, interest to join your lab. Dear Professor something, having read through your research experience and your papers on the department of this so so so, University of this so 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 so, I would love to work under your supervision for my PhD or master program for 2023. Now, after that, you now go about who you are. I am a graduate of this also university with this also GP. I have numbers of these publications. I have GRE, I have TOEFL, I have Duolingo, I have Google. You state your strength. Then your paper on this also or your research work on this also so attract my interest, and I would love to explore it greatly in your lab because we have similar research experience. Then after that, we talk about how teachable you are. I am teachable and I'm ready to learn. Through learning from you, I'll be able to publish and present and also discover patentable or patentable, what you can make patent of. So then you'll be looking forward to hear from me. Attach your CV or transcript or anything. We get so not all professors will now reply. But as far as the graduate coordinator have replied you and you have all, you can apply. Are you getting me? So now, if professor reply you, professor will indicate what he want. He will still tell you to apply. That is why I tell you that you have to ask the graduate coordinator first before you apply. So when you have all the needful thing, you go ahead to apply. Or to go ahead to mail the professor then before you apply. So to answer your second question, to your third question, you can do PhD directly without a master with the number of publications that you have and your research experience. What, as far as what I know, is about PhD is about the skills that you have. What is the skills that you have? What is the level of your experience in that skills? And are you teachable? Because they have to teach you everything from scratch again. So when you are teachable, then if you are teachable, you will be able to publish because you yield to corrections and mistakes. That is all.
Oh, thank you so much. I think we've gone past our time. So other than I would suggest that it be asked on the Telegram page, and then other promises that I've made, I will also share the material in the group. So before we do that, I want to appreciate our resource person. Uh, is my Oga at the top, and we are always proud to be associated with him for his great achievements and everything. So regarding the mentorship, he is very very much. He makes himself available to mentor and put our students through, and even those who, as long as they are willing to learn. I mentioned it
How
at the start
Sorry
of
for cutting you short.
that.
Sorry for cutting you short. He can mentor us in what areas? Let's know his area of specialization so that we can either apply if we are in his own field or in line of study. You know.
I would answer that question. He doesn't need to answer. As long as you are in research, just forget about it. He will mentor you. Um, actually, it's good you asked that question. So I was actually going to elaborate on that. If you started with us from the beginning of the class, I made mention of the fact that the latest publication that we had in Nature, it was as a result of his idea that he gave me an assignment that oh yeah, Oga, I will write this thing. Even though I postponed. So I eventually wrote gave him the assignment, and luckily the paper was accepted in Nature. That is the fact that once you are teachable, like once you are teachable, you would break a lot of barriers. Ladies and gentlemen, this single paper that you are looking at, I'm talking about, it has won me a lot of special seats in my laboratory, even in my university, all the whole of KNU. KNU is one of the best universities in Korea. So that's the benefit of once you are ready to put yourself to learn, irrespective of who or where. Don't worry about you breaking barriers. You break barriers and you do well for yourself. My guy is not, he's not the one that is in the first author, and I'm the one carrying all the glory. Like, nature, they will not know that my clinic has something in the back. So that's one aspect. So he can mentor you in a lot as long as you're in research. So you can reach out to him. Someone has asked for his number. Well, he's still a bachelor, but um, so that one too is also there. Just kidding. So I will share his contact with us on the. Do I have the permission, sir?
Yeah, why not? Yeah,
Okay,
sure.
so
Definitely.
I will share his contact with us on the Telegram group, and we can reach out to him and also ask the required questions. So I would try. I want us to call it a day here. Okay, I'll also share that of Dr. Sudia too, for us to reach out to this mentor. So I'm glad that God has used me as a figurehead to organize these sessions. At first, I was very confident of the fact that those that we will be bringing and benefiting from these sessions are people of timber and caliber, and I'm very glad that they've done even beyond expectation all through. So without much ado, I would like to call it a day here. So I want to thank every one of us for being here. Tomorrow, God willing, I want us to be here also in large number, and so that we can benefit from tomorrow's sessions. We pray God will grant us the grace and the benefit of being a beneficiary of these great sessions. There is a certificate after this program. I think now the certificate should be made mandatory, because you can't benefit from all this and just let it go like that without having something to show for it. People have been here and they've committed their time and money for this endeavor. So thank you so much for being here. Really appreciate your
Before you go, sir, it will be a great pleasure if you drop your contacts, because I did my IT in molecular biology, and I would like to, from there explore into bioinformatics as well.
okay, sir. Okay, sir. My contact, if you're on the WhatsApp group, I think my contact is, of course, you have my contact.
WhatsApp group, sir.
Okay, I'll drop it on the Telegram group.
I already have experience in the DNA extraction, ELISA, SDS page, and everything. So I would like to explore more and know more under your mentorship on that.
Thank you. So that's good. That's good. So thank you so much for everyone. The contributors, Mr. Effiong, Mr. Sunday, Dr. Patricia, Dr. Adebi Dokas, and Dr. Laleko, everybody, Dr. Laleko,
I'm a student. I'm not a doctor. Olale, I'm a student.
everybody. Thank you so much for being a part. See you tomorrow by 10 a.m. Where we'll be talking about cytoscape GU and the introduction to 16S microbial diversity analysis. Thank you so much. We can pen the questions to tomorrow. We can start the session tomorrow with answering the questions that have not been asked here. Thank you so much. Thank you so much, everybody. So see you tomorrow and have a nice weekend.
Thank you, sir. Bye, sir.
Good night. Let me stop
Yeah.
and share.